Hello.
Okay. No, he is not there. Instead of that, Ishwari. Instead of Venkatesh, Venkatesh. No, Venkatesh with Ishwari.
Good morning. Good morning. No. Ah. Good morning to all and welcome to the third day, half a day of what I'm sure is going to be very exciting lectures. And we end the day with a special lecture. So please do stay on till the special lecture. And then if you don't stay on, you won't get lunch. Okay? So we'll take attendance. Okay, so without further ado, I invite Professor Krishna Kumar from RRI Bangalore to take us through till the tea break. Please.
Thank you. Good morning to all. So the first speaker of our session is Professor B. Senthil Kumaran from School of Life Sciences, University of Hyderabad. He works in the area of fish reproductive biology and molecular endocrinology. He is a recipient of DBT Tata Innovation Fellowship, visiting STA and JSPS Fellowship from Government of Japan, Global Ambassador Fellowship from University of Saskatchewan and JC Bose Fellowship. He is a fellow of National Academy of Agriculture Sciences and fellow of all three major science academies in India. So he'll be talking about brain sex differentiation in bony fishes. To you.
Thank you, Chairperson. At the outset, I would like to thank Indian Academy of Sciences for giving me a chance to present my work with this august audience. And today I'll be speaking on brain sex differentiation in fishes. I put on quote because the idea for brain sex differentiation in fishes mostly came from our laboratory. So we were trying to prove that the brain also play a role in the process of sex differentiation. Usually gonads play a role. So we have proposed an hypothesis, and most of our findings, maybe end of my lectures, maybe you may get convinced partially or fully that there is a possibility for brain sex differentiation. First of all, what is brain sex differentiation? Most animals, the gonads dictate brain, and based on that, the sex hormones coming from the gonad have an influence on brain, so that we can have a situation of a male and female brain. And when you talk of brain sex differences, naturally you will see whether it's gender specific. So certainly it is gender specific, because most of the times we always feel that multitasking, but men cannot do multitasking. If you see men, they have isolated one task, they'll be thinking on that and they won't do beyond that. I'm very sure actually. Most of you know that, how many of you know that you have called your son or daughter about on the exam day? Most of the time I fail, my wife tease me, asks ordinary multitasking, because the white matter in the females are more. We have a gray matter, so we are naturally on one focus. And then men also have a different problems. We have an amygdala, because of that we are more aggressive. So either we fight on a situation or we will escape from that fight, flight, we will flight, that's all. But we don't have the intermittent thing. But the females have this intermittent also. They will fight, they will flight also, they will join with that also. So we never forget. Sometimes we fight means we never forget. With female, sometimes they will forget it and they will get along. You can see, for example, in olden days, even nowadays also, we wait for water. They will line in queue, and after that, what will happen? Once that during the queue they will be very strict. After that, they may share food also in the back home. So that way, also the hormonal response behavior is somewhat different. And also, so that's what I'm saying, that a woman can do many things at one point, but men will always get puzzled whenever many things are coming in hand. Especially when you become old, you can see grandma will dictate grandfather actually. So always it happens actually. So even in my case, my grandpa is MA English literature. My grandma is only 3rd standard, but she will dictate him what to do. Okay, I have seen on my own eyes. So I agree with that. Maybe when I was very young, I could not understand what is happening, but now I know this is happening because of the differences Almighty has given us. And then also brain also participate in several of these monominds, give different kinds of inputs, and especially for men and women, they don't have a similar parameters. Even we have a, like right, left also, you can see there are a lot of differences always happen in the human as well as mammalian brain. And you can see number of these hormones behave differently in men and women. You can see, for example, if you take prolactin, I have not mentioned here, prolactin is a classical example. Prolactin is a hormone which is lactogenic. You can see the differences in prolactin hormone when female is not pregnant, and then female when pregnant, then the levels of hormone goes high. Instantly, if you see the single father who did not get married or continued to live a single father, they have quite a bit high level of prolactin, with the normal woman don't have actually. So it will be in between the lactating woman versus normal woman. Prolactin levels are very high in single father. So there are differences in hormone based on the behavior also, and also based on the sex also. And then you can see some of these peptides which are coming from the posterior pituitary, especially oxytocin and vasopressin. These are 9 amino acid nanopeptides. They behave differently also. For example, if we take the Pereira-Oles, they bond for life actually. So they will be husband-wife forever. And if they get separated, the male will have a solitary, similarly female. But whereas the same kind of Montagne, they don't behave like that actually. So there are plenty of examples coming in the mammalian literature as well as human, that there is a perfect brain sex dimorphism. And also the feedback regulation, if you see, the female has got both positive and negative feedback, which is not there in the men. Men always have one negative feedback, that's only negative. And because of that, the female is most advanced or evolved organism in terms of evolution actually. So because of they have the cyclic center in the brain, because of the female is again evolved. But when we have all these literature, so we thought, why not in fish will have such kind of a brain sex dimorphism? That was our question actually. So we started this journey maybe 15, 16 years before, that there should be some differences, because we see some examples. I can show some examples why we believe that. So on the other hand, like a negative and positive, fish do not have a positive feedback. They're all negative actually, whether you take male or female, there's only negative feedback. This was a big disappointment for us, that to go about it actually, because there is a positive negative for the mammals, but we don't have. But still we decided to work on fish to understand whether there is any kind of gender difference we are seeing actually. So that was our question. And then this question is a little bit important, because some of the studies shows that we can change fish from male to female. Changing fish means I am saying that changing the sex of the fish functionally. For example, if the male became female, they'll start to produce ovary, eggs, everything will come there. Similarly, if the female becomes male, they will also become producing sperm. So it's a fully functional sex reversal. The fishes are the only vertebrates which are called champions of sex reversal. When you come to frog, only one way is possible, female will become male. And in reptiles, temperature can change. But once becoming a sex is established, changing can be done only in fish. And we can either do naturally or we can do with the hormones. And then gonadal transdifferentiation always happens first than the CNS system actually. So in this case, always gonad will change first and then brain will follow that actually. And these are some of the examples which we see, Protandrus and Protogynous. This fish lives as a male for 1 year, Then second and third year, they will become female because the male is bulky. So naturally, the male is get harvested. Then the biggest, male will become female. Similarly, here also, if you see the female will get harvested, and then because of that, one of the female will become male, and then the school is continued actually. So this is a natural way of doing it. We can also stimulate this by treating hormones early times. So if you treat with estradiol, you can have an XY female. Similarly, if you treat with XX, treat with androgens like methyl testosterone, then you can have an XX male. So the alphabet of chromosome is just an alphabet. So we don't call it as a significant. So there is genetic sex, hormonal sex, environmental sex, plenty of these are there. And then, so this is what I was telling, that you can change it at the early stage. You can change it to the either sex, whatever you want. And then here also the gonadal differentiation takes place first compared to brain. And there are some other fishes which is what giving us an example that certainly brain sex difference is there. These are serial sex changes. What happens if you have a pool of fish, and then one male will be there and 3 females will be there. So the male will do the parental care. And if you remove the male, the biggest female will become a male actually. Then if you bring back that male again to that area, or if you taken in an arbitrary lab setup, then if you take that male and then bring back that male, the biggest female which changed sex will revert back. So these are called serial sex changes. All these happen in 1 week time. Okay, so here you can see the CNS changes can be in upper hand because the male shows the aggressive behavior, takes food first and prepare nest for egg laying. So it will bring the female to egg lay, and then it will shed the milk, and then it will protect the young ones. So you can see that behavior from the very beginning. So this gives us some kind of an interesting idea that certainly brain will show some response. So it is always showing that kind of a behavior means there is some response will come brain. So we thought that it's worth investigating. So these are some of the fishes which we essential models, which is coral reef fish, like our the clownfish. They live in a small school. So then that one male will be 3 females. So imagine if one more male comes, then the male has to fight out which male will be there, like a lion's pride. So this is one example. Another is Thalassemia duprae, which is again a coral reef Hawaiian fish, a little bigger than the one which we saw in Thalassemia duprae. And then there is also other fish models. But unfortunately, I didn't have a chance to work on these models because you have to scuba dive and catch fish. I never even swim also, so it's very difficult for me to scuba dive. But somebody told me that swimming is not needed. You can scuba dive. Still, I didn't have the courage to learn that. So I thought, why not we work on fish models which are available with the genetic sex populations. We have a Nile tilapia. We started our work way back in Japan with XXXY populations. We know that it's become XX and XY. So it was a very good model. And then later on in India, I started working on these fishes, which are domestically available for me, and also which is a very important fish in terms of food fish in India. So most of my work first started with Garipinus, which is an African catfish, which was not accepted to research by DBT, but SERB always, DST always support that. But later on, we switched to Batrachus, which is an Indian catfish. So we rear this fish and also have collaboration with that one. So starting with tilapia. Tilapia is a very good model because it's a fortnight breeder. So we have an XXXY population. So we thought to work with this first, and also I have a chance to have the success of XXXY population. So we started this work. And the life cycle of Garipinus is 1 year actually. So you have to wait for a year to do this work, and we don't have any XXXY population. Batrachus also similar actually. So first we decide how to proceed this topic actually. So we thought first to go with the GNRH, and before that we also worked on some of the amines we also worked. So I will come for the GNRH and then go back for the amines actually. So GNRH is an important releasing hormone set right the gonadotrophic axis. So we started analyzing the GNRH protein, both XX, XY population. You can see this XY male, it appears very early, somewhere around 7 days, and disappears and then comes back again. On the other hand, in the female, you can see the appears around 15 days once the gonadal differentiation happens, and then it gets stabilized. So there is subtle difference actually. So it means that there is a GNRH coming up and then goes off and then comes again. But on the other hand, female, it comes off little later after everything is established. So one area we thought there is some differences. And then we thought there is also something in gonadotrophins, but we didn't get any changes in gonadotrophin. So GnRH difference, dimorphic difference in terms of appearance was first clue. Then we thought to go for amines, and amines is very important because they set right the GnRH axis, especially serotonergic and catecholaminergic system. And we got some information before that if you block serotonin, which mimics like estradiol, especially early time. So this was a kind of a causative response. You block serotonin in brain, and then it happens in terms of gonad also. So this was a small study available before we started. So we thought to clone this rate-limiting enzymes, that's tryptophan hydroxylase, and see what happens to this enzyme, because certainly there should be a difference between male and female. So we cloned this, and then we found that the early expression XX and XY only have this particular enzymes. In 0 day, you can see some expression, but because we use the whole larva or whole fry. But fry day onwards, we have checked only the brain region, and you can see the females does not have, but the male do have extraordinary amount of TPH with the quantitative PCR in terms of RNA. And then we want to compare this ovary and testis also, because they express little bit serotonin. So then we found that it is not showing any dimorphism, only brain dimorphism is seen. Then we wanted to localize this in the transcripts. We localized, again, nothing is there in the male, but nothing in there in the female, but male have got plenty of transcripts of tryptophan hydroxylase, which is the serotonin synthesizing enzymes. Then we also see the protein. Again, the XY male have got a specific expression of protein is found in the XY male, but not in the XX female actually. So this gives an idea that clear-cut sexual dimorphism. Then after 30 days, this does not happen. Then we went on to Clarice batragus or Clarice gariepinus also we checked, and then we found that since we don't have an XXXY population, but then you can see around 50 and 75 where there is a differentiation takes place. Again, the overexpression is seen only in male, and then we also see the protein levels especially in the preoptic area. Everywhere the male shows a very high expression actually. So which tells you that the TPH is selectively found in male during early time, but not in female actually. Then we also analyzed the precursors and the metabolites which are coming from the serotonin. Again, serotonin TPH. Then you can see the serotonin levels and 5-HTP, everything is going high in the male. So the enzyme and the products, everything are at higher levels in the males. And then we also reversed by changing sex. So we did an MT treatment. MT means methyl testosterone. We also treated with estradiol. Instead of estradiol, we put PCPA because it mimics like estradiol. You can see that in the PCPA, whenever you give PCPA, that means estradiol level goes down. So that tells you that the tryptophan hydroxylase is towards male, but not towards female. That means there is a selective elevation of serotonin in the males, but not in the females. So that explains me that the GnRH in the early time, because of this serotonin, it appears and then goes off and again stabilizes. So this was the first finding which we did, and we are first to show the brain sex dimorphism in terms of serotonin and the enzyme, and which is a kind of a causative response, not consequence. But all other studies later on we could get only consequence. And then in the females, we started working on the candidate gene brain aromatase, because females have got a lot of aromatase activity overall in the brain. And then we also could see the expression of both the aromatase always found high in the female, but not in the male. And then you can also see the expression is pretty high, and also the transcript which promotes the aromatase enzyme also very high in the females actually. So then we also checked the catecholaminergic system, which is another system which is operating in GnRH axis. So you can see all of them, especially the enzyme tyrosine hydroxylase, which is a rate-limiting enzyme for catecholaminergic synthesis. All of them shows very high expression in the females, but not in the males again. So that tells you that the serotonergic system is there for the males, whereas the catecholaminergic system is there for the females. But once the sex is established, it becomes normal actually. So the difference only available during the early time when there is a differentiation takes place. And then we also measured the metabolites and also the products that are coming from that. Everything is showing very high levels. And then you can see the female always tend to show, and then when you reverse with the estradiol, you can see the estradiol promotes. That means female means you have a lot of catecholamines. It promotes. When you give the methyl testosterone, it doesn't promote actually. So then we have we also studied the GDNF family, which is also a dopaminergic system establishing. that also is towards female, very high. And then NPY, which is another peptide which is stimulating GnRH, also very high in the females, but not in the males actually during early development. So number of markers we have listed, and then we have found out that most of them for the females, but only serotonergic and dopamine system, this is TPH is for the males actually. And then we also tried to, put these sex steroids reversibly, like putting estrogen in the male and the testosterone in the female, and we have used, so we have used an osmotic pump.
Through the reliable process of osmotic displacement. Here's how it works. Howsat osmotic pumps consist of 3 concentric layers. The inner drug reservoir, the osmotic layer, and the outer rate-controlling semi-permeable membrane. The reservoir within the core of the pump is filled with the test solution to be delivered. When the pump is implanted, water is absorbed through the outer membrane. The water entering the pump expands the osmotic layer, compressing the flexible impermeable reservoir. This enables the test solution to be released through the exit port at a controlled, predetermined rate.
So we used the osmotic pump to release estradiol and testosterone selectively, and then we also found the changes, whatever transcripts we have shown, that especially it is towards female and male. You can see the changes that is happening when you mismatch it. So that also again confirms that some of these markers which we identified are showing sexual dimorphism. And then we also showed at the level of brain with the immunohistochemistry, whatever we have found out that mismatch because of these hormones, we can see the changes, whatever markers we have described. So overall, we have found several markers for female side, but for the male side, we could always get only the serotonergic system at early development, which is showing a cause response. Remaining all of them is a consequent response, re-establishing that the gonadal will dictate the brain in terms of brain sex differentiation actually. So we also established a neurodegeneration model with this kind of an idea. And also we are now working on creating neurodegenerative models, especially using annual breeders. Usually you have daily breeders like zebrafish is a good model, but we are now establishing annual breeder models which are working very well for neurodegeneration. So all my studies over the time now helps me to understand this behavior pattern, especially neurodegeneration, which is different between male and female actually. So these are some of the publications and my research team which are presently working with me. And then most of this work was done by the postdoc, with my Mamta, and then my wife Sudha also did along with late Professor Aparna Datta. She was with her actually as a PDF. And I'm thanking late Professor Aparna Datta for a lot of intellectual exchanges between us, made me to grow on this topic actually. Thank you very much. I'll be very happy to answer more.
Questions?
Yes, sir, but in catfish we don't see. Tilapia we do morphological differences. The male is little more heavily pigmented, but the female is little light. Yes, sir. Yes, because they show that aggressive pattern, and which will be very much seen in this coral reef fish actually. But this kind of domesticated food fish shows very less changes, but this the ornamental fishes shows fantastic changes sort of completely. You can even while changing itself, you can say that this is going to become actually. So you can see that. And then interestingly, if you put them in a two different glass, people always say that the fish have got less eyesight, but still in the glass itself, if the male is there, it won't change sex. So that was very interesting observation. One of my Japanese colleague worked on that, but he did not continue. But he showed me that put the Trimacanave male in a glass with the same glass. When that male is there, it won't change. But people always say the eyesight is poor. It is not in the next aquarium, but still it didn't change. Only marine fishes have this phenomenon. So if you want to work in a freshwater fish, then you have to simulate, like by hormones. You can only do. Natural behavior is very less in freshwater fishes actually.
So you said in the beginning here. Yeah.
When you're doing the experiments, how many fish do you?
Usually we used to more than 5 or 10 actually. So because we used to put all the fish, for example, estradiol, we used to have 100 fish. So when you put estradiol to them, so almost like a 95% will reverse actually in estradiol, especially in catfish. But in the case of testosterone, interestingly, testosterone is less effective than estrogen, at least in catfish, unlike other fishes, where again we will use around 100. But when we do for sampling, always 5 to 6 for each of the experiment actually.
For this development, is there any stage which is, because it's a transition, you said yes or no. So it just transform, but is there any stage which is in between?
It's a very good question, because some of the fish will be requiring the change can happen only early. Some of them will change early and juvenile, some of them in adult. So the work model which I am using, like tilapia or catfish, can only change in the early development. So it does not show any. That's why when I mismatch the steroids, the molecules will show change, but it cannot become a female or male. Only thing is they will have a disturbance in terms of fertility that may have, but they don't change sex once they become adult. They are very dominant when they become adult. But when you are young, you can change. But certain fishes, like I told, correlative fishes, even adult is succumb to change. So each fish species have got a different window, but on each window they can change completely.
The XY male in the on the Y chromosome, there must be genes that are expressing. When the XY male start behaving like a female, what happens to those genes on the X chromosome? Do they stop expressing? On the Y chromosome, do they stop expressing?
So answer is available with Medaka. We found a DMY triplicate of the DMRT1. It has been established as a sex determining gene. Later on, AMH also found. So whenever you treat with this estradiol to those animals, the aromatase expression shoots up. Though aromatase in autosome, it shoots up and it down-regulates the DMY expression. So that's why the sex determining gene Y chromosome can be easily regulated down with the hormones.
Does that happen in all cells or only in gonadal cells?
Only in gonadal cells. But instantly, this kind of work is available only with the few fish species where sex determining gene have been identified. For example, zebrafish, there is no sex determining gene till date. So the answer is we don't know how it is. But certainly, XY is a mere alphabet for them.
Okay, I guess any more further, any other question we can take up during the break. Let's proceed with the next speaker. So there is a change in the second speaker. Instead of Vikas Kumar is going to be Professor Ishar Institute of Astrophysics, Bangalore. So Professor Reddy is a senior professor, is a dean and
Yeah, Mr. Chuda.
7 from IIA, and after 6 years of postdoctoral work, he has moved back to IIA Bangalore as faculty. His research interests mainly involve the chemical tagging of stars to understand star evolution, nucleosynthesis aspects, and the evolution of the galaxies. His work on the Milky Way galaxy is well known for its chemical decomposition of the galactic disk into thin and thick disks, and for discovering the helium flash as a source of high lithium abundance among giants. He is also spearheading India's participation in the 30-meter telescope International Observatory as a program director of the India TMT Center. He was elected as a fellow in 2023.
Thank you very much for the introduction. Good to see the spirit of so many people in the morning after a very nice dinner last night. So today I'm going to talk about the lithium, and particularly related to its evolution in red giants, because the stars are basically sinks of lithium, but then there is a puzzle which was there for more than 4 decades, and then we pursued that with my students So then we found some very interesting results. So basically, I'm going to talk about lithium, and then you must be all knowing about what is the lithium. And the lithium is, third element in the periodic table. And then basically, we look at high-resolution spectra, and then analyze them, and then quantify the lithium amount with respect to hydrogen. So much of the credit goes to my students. Bharat Kumar is a faculty now at IIA, and Raghubar is in China, and Deepak is in Aries. Anohita Malik, she is currently doing with me PhD on this subject. So as you know that the lithium, along with hydrogen and helium, has a primordial origin, and the basic reactions, you know that, and that's the beryllium plus neutron, and which gives the 7 lithium. This is in the Big Bang nucleosynthesis. These are the 12 reactions which define that. And then lithium normally we expressed in the stellar abundances relative to hydrogen and normalized to 12, so that then we have a much better understanding number. So on logarithmic scale, I just give you the number. You can see the primordial lithium, the current, the state-of-the-art BBN models, and then with certain parameters like, baryon to the photon to baryon ratios, which came from Planck and things like that. Those are some of the experimental values. This is the best value now. We have 2.7 primordial value. And then on the other hand, we can see on Earth, that's a 3.2, which is similar to, came from natal clouds, like interstellar medium, that's currently that's amount. And then on the other hand, on the sun, we can see it's a main sequence star, which is almost like, 2 orders of magnitude less than what you see on Earth, or even in the ISM, is 100 times less than. That's what it is, the logarithmic scale. So the cosmic lithium problem, we can see the prediction of light elements with eta as a free parameter. You can see here, this is the helium one, and then the deuterium, and then 3He. They all came from the Big Bang nucleosynthesis. And this is the lithium. And then the baryonic to photon ratio is a free parameter here. Once that parameter is fixed, and then you can see that this is the one where the observations show, and then the helium observations are perfectly matching, and then the deuterium are perfectly matching, helium match. On the other hand, there is an issue with 7 lithium and the BBN, the predictions. And that's called the cosmic lithium problem, which I'm not going to too much talk about that. And then you can see as a factor of 4, the difference, which is quite significant. That is still a problem. So what I'm going to talk about something related to lithium evolution in the galaxy. It doesn't matter which I can take, either BBN model, or there is an observations which we all believe that that's a primordial value, because many of the old very metal poor stars have a very less scatter across large range of metallicity that we think is a primordial value. Actually, that is in the logarithmic scale, the 2, which is a factor of 10 less than what is kind of, a factor of 6 less than the BBN model. On the other hand, if you take, the BBN as a 2.7 or speed plate as a 2, and then you can see the ISM, the the lithium is 3.3. So basically it is increased, either whichever you take it, either observational or BBN model. If you take observations, it's a factor of 10 more. If you take a BBN, it's a factor of 4. So it doesn't matter which you take, the lithium is enhanced over the years from the Big Bang to the current value. So that's what I just want to say. And what are the known sources? Known sources, one of the Spallation reactions, that galactic cosmic rays. And then, of course, the stars, there are, the people suggested stars also can produce. The major thing is stars do produce, but then bringing them out is extremely difficult because lithium get destroyed very fast in the stars. And lithium synthesis and depletion of stars, you can see the problem here is that lithium can be produced within the hydrogen burning cell, and then bringing them is extremely difficult because the beryllium get produced at something 10 power 7K, and then that can immediately go to the lithium here, But the problem is then the lithium get destroyed at much lower temperature. Bringing this beryllium, you have to bring the beryllium to outer atmosphere where it is cooler, and then that can transform into the lithium. Then you have to bring to the photosphere. That is the tricky. Those are time versus temperatures. That is an issue. That's the way the models we didn't have. So everybody thought the lithium stars are sinks of lithium. So little bit on this, you can see these are the main sequence stars basically. In astronomy, basically, if you are doing stars, everything you represent with called a Hertzsprung-Russell diagram. This is the luminosity, and this is the temperature. Is a proxy to the temperature. Is a color. You can also. So most of the stars stay here much longer time, and then the stars evolve. This is sun, then become subgiant, and then a lot of convection happens, and then the stars go in the red giant branch, and then they come down. This is called a clump, and then again they go back here. This is called asymptotic giant branch stars, depending on mass of the stars and compositions. They go here and then end up as white dwarfs, mostly in the low mass stars. And these are the chemical reactions which happen. The 4 hydrogen nuclei fuse together and become the helium. And through these kind of reactions, in between, in the stars, what happens is now when the stars is going up, and basically you decrease in carbon enormously because the 12C goes into 13C. And then the 12C by 13C increases, that is 13C, because 13C increases. And then increase in the nitrogen because of this one, the 14 nitrogen increases. But then the total C plus N is constant here because you don't have a fresh carbon or fresh nitrogen because you are not fusing helium triple alpha reactions. So this is the catalyst basically to fuse 4 hydrogen atoms into 4 the helium nuclei. So what the standard models say, even the current state of the art models, and normally if you assume and the main sequence stars something like ISM value, the way it is, and then once you go to the red giant branch, and then 30 to 60 times less lithium. That's what all the models tell us, depending on the mass. And then the convection, because of the convection, and then you have a ratio of 12C by 13C on the main sequence 90, that becomes 15 to 30 when you go to the red giant branch stars. Because of this convection reaches the hydrogen burning cell and brings the byproducts of hydrogen burning cell onto the photosphere. That's why you see these numbers. But actually, these numbers are much, much, much lesser than what the models even predict. So that means, for example, when I make the observations of Arcturus, a typical K giant, a bright giant, and you see the lithium is much less than what even the predictions say. And similarly, when 12C by 13C in many of the stars, most of the stars reaches equilibrium value. So there is some problem there within these models. Again, lithium is not necessarily the ISM value on the main sequence. Many stars differ the value. For example, the sun has much, much less lithium than what is the ISM. That itself we don't know whether it is during the pre-main sequence itself is got destroyed quite a bit. So little bit about, I want to just talk about something details here. I don't want to bore with that. So while stars are going, there are 3 main incidents happen for the star like a sun. One is here, it's called the first dead jump. Here is the place where a lot of hydrogen burning products come onto the surface. And there is another one, it's a small thing. When you make observations, you understand that. This is called luminosity bump. And here, stars stay for longer, maybe 10 to 100,000 years. That's why when you make observations of similar age, and then you can get stars get clustered here. So they just go up and then come back. I don't want to go into details. And then after that, stars continue to go up, and a lot of convection, very deep convection. And then the entire phase gets terminated here. All the low mass stars undergo, they develop degenerate helium core, electron degenerate core at the center. And then a thermal runaway happens here because of the continued pumping of helium ash onto the core through hydrogen burning cell. So here is the one the helium ignition starts, and then since the helium ignition starts, there is a called, adiabatic expansion happens, and then suddenly the gravitation overcomes the radiation pressure, because hydrogen burning cell energy comes down, and then the radiation pressure is lesser, and then it shrinks. And then the star of something like 50 to 70 solar radii becomes 15 to 20, and then suddenly it drops here, and then this is called a red clump stars. There they stay for 100 million years, and then people say, the models indicate, suggest that this entire conversion of degenerate core into helium core burning at the center takes around 2 million years, but we don't know exact stages. This is just I wanted to give you, though the formulated this entire degenerate core is to 1962, we don't have any observational evidence exactly that is one is happening. There's no observational evidence because the degenerate core, that the helium flash, first helium flash, the prediction is it produces the energy of 1 billion times more than the sun. But where the entire energy is going, we don't know, okay, the fraction of whatever. So the most of the energy goes to remove the electron degeneracy there, and the rest only partially goes into lifting the hydrogen burning cell outside, and that's where it gets cools, and then the star shrinks, and then because of it shrinks, and then the luminosity also falls here, size and luminosity both. As you can see almost like 1.5 in the logarithmic scale, 20 to 30 times less luminous stars. I'm not going to. So these are observations now. What we are looking at it is observations. We can see now, this is the lithium I'm plotting, and then this is again temperature V minus V, I give the proxy to the temperature color. And then you can see all this, on the main sequence, these are very metal-poot stars. You can see, I said the observationally speed plateau is the primordial value around 2.2 here. And then the first dead jump happens, and then suddenly lithium comes down here, extremely. For some time, the convective cell recedes from the hydrogen burning cell because of the temporary discontinuity with the hydrogen protons availability. Then something is happening here. You can see this was not there in the standard models. And then you can see the cluster of few stars here. Normally, you can see many more stars. This is exactly the place that is called the luminosity bump is happening. Stars are reversing. And this is the where it looks like it seems hydrogen burning cell moves upwards, and then it accesses a lot of protons from the cooler regions in the convective envelope, and star for temporarily cools down in the energy wise, and then the size also decreases, and then suddenly it falls down here. That's why they stay here for a longer time. On the other hand, this is the place where extremely less lithium, and 12C/13 is so low. And this is where now recently models that extra mixing is happening. So there are various models for this, how the extra mixing is happening. So basically now you should see observations and theory says you should not see lithium. The basically is mostly get destroyed. So that's what it all says. You see the observations clearly says, and then the theory also says the similar. But then the puzzle is, in 1982, 4 years ago, you can see this is one of the lithium normal star, KJN, and then you can see one of the lithium line, that is 6707, a resonance line. You can see a very strong line, and which has abundance of almost 3.2, that is interstellar value, and which is almost like a 100 times more than what even nominal theoretical models predict, and much more than average lithium abundances in RGB stars, maybe 1,000 times. So that was but then there was not much follow-up on that. And then people thought there's something is happening. So there was a review in 20 years ago, and then they collected various in the literature, and then they had something like 2 of them. Again, they are very near the bump where extra mixing is happening, which is depleting the lithium. So just a couple of super lithium-rich stars, and then again, then a lot of theoretical models came up about how you can produce lithium during the luminosity bump. So we asked the question, okay, before that. So I was doing something else. So, and then this happened to be in one of my samples. Everything looks like perfectly normal star. There is nothing abnormal about this, except the lithium line. So then we looked at it, and then where it is, what the spectral classification is, a completely red giant. So I looked at it carefully on these things, and then it has infrared axis. And then, of course, there's a slightly larger velocity, rotational velocity. And that time, people were talking about, planets already discovered. because planets also plunge into the stars. And then planets induce angular momentum. And then since planets are so small, the lithium is not destroyed by the time the Earth, then that requires a large amount of lithium. So maybe, large amount of mass has to thrown into the convective envelope to see this increase. So there is an infrared excess is there. The plunging gives you some kind of infrared excess. The dust throws out, and then there is a higher velocity gives you, angular momentum. So that perfectly fits. So, but then we were not sure about that. Then we looked more carefully. So these are the theories earlier. No, lithium was not basically getting destroyed. It is preserved from the ISM. But that was not, that was ruled out because there's a huge convection in the stars. And then planetary white dwarf engulfment, there's a planet engulfment. So this is one of the theories still there, and then nucleosynthesis and mixing inside the stars. And then we took up a survey. I had a very good student, the Kumar, and then getting this high resolution spectra is extremely difficult. It takes each star, even now, star by star we had to do. It takes 30 minutes to 1 hour on bigger telescopes. So since we didn't have the spectrographs here, high resolution that time, and then what we did was, let us do with the low resolution how to get these things. So we chose some 2,000 stars spanning from the bottom of this evolution, like a first dead jump, all the way to the up, and then thought of looking at these number of stars here. So amazingly so this is a technique we used because this is the first time we used. That time there was no surveys. We used, there is one lithium line and with calcium lines with extremely low resolution spectra. And then we use the ratios because these lines are insensitive to the temperature because both have a lower excitation potential as a 0. And then we look at the only similar metallicity because the calcium is the metal and then lithium doesn't care. It's not a function of anything, It is sensitive to various things. So we used this technique, and then what we discovered was we found almost 6 or 7 super lithium rich stars and then some of the lithium rich stars. What you found is, amazingly, at particular luminosity, this is a peaking. All the higher luminosities are something 1.6. The problem came is, then we made a suggestion, this cannot happen at the extra mixing is happening, luminosity bump, because this is the same place where extra mixing is happening, lithium is getting destroyed, and the same place you cannot bring the lithium up, such high amounts. And then even if you bring it, so so basically you can't bring it. This is the hypothesis we made in that paper, 2011 with Mike Kumaran. Then again we followed up, but then we don't know how to solve that problem. How can a place which was identified for very low lithium and then can be also source for the high lithium, that's what. Getting the spectra, you can solve this problem if I have a bigger telescope, better resolution spectrographs. I can go to much globular clusters, very faint globular clusters, I can solve this problem, but then it is extremely difficult. Then the boom came because astroseismology, that Kepler data released, and then they could see the difference between the stars, between RGB stars and RC stars. What I'm saying is the RC stars is helium core burning stars. That is after the helium flash. The stars are after the helium flash. RGB stars are on the way, is ascending the RGB phase. In this case, helium is inner to core. Only hydrogen is burning. And then in this case, the helium is also burning at the center. Hydrogen is also burning in the shell. Okay? So you can nicely differentiate using the G modes and then P modes, the frequency versus spacing between oscillations. And this was a very nice thing. So what it solved the problem was, suddenly we found 2 stars which are unambiguously, these are red clump stars, and they have a super lithium rich. This is one of the first evidence we got. There is something which is, nothing is happening on the red giant branch. There is something after helium flash. Still we didn't know, really the helium flash is the one which is causing this entire thing. Then another survey we took, this time we used some survey. That survey became available in 2016 or 17 in the Chinese Lamos. Again, low resolution survey, large number of stars with the fiber. And we found 30 of stars really very high super lithium rich. None of them were on the red giant branch. So we continue with that, and then this is the first evidence we published in Nature Astronomy, and then so the lithium enhancement is ubiquitous among red clump stars. And then we also demonstrated the lithium only depletes on RGB phase. So that was one of the key. And then, so far in the last 40 years, the nomenclature for any lithium-rich stars more than 1.5 doesn't apply here. It is actually minus 0.9 dex. Any star more than that, it is a lithium-rich. So I'm not going to go. So we even set the evidence too, and then we set the upper limits within that time. The ignition phase started, maybe 1.5 million years or something, and then the entire thing is happening. So these are some of the more, these are the flashes, which you can see the major flash, and then the next flash, next flash. I'm done. So based on this, we put some time limits on this. Okay, I'm not going to the evidence 3, but then I just wanted to say that now the helium flash is the one which is producing lithium, but now inspired by this, now more than a dozen papers, theoretical papers came out, and then we had to see which one really is working on that. So, and of course we need to actually come up with some quantification how much this lithium is contributing to the galactic chemical evolution, so that we only. Another thing is none of these stars show infrared excess, so we don't know what's happening there. So thank you very much.
No questions, but I would like to ask you, when you are modeling all these things, how accurately you have this nuclear reaction cross-section which goes into the modeling for all these?
BBN modeling we don't do. Okay. We just take them granted. Of course, there are a lot of uncertainties in those reactions. For example, a lot of people say now 2.7 must be much less if you allow that beryllium into the beryllium reaction. So it may not be 95% or even 85% if you reduce it, that BBN things comes down. So
what I meant is that there is no unpleasant surprises coming from basic nuclear data into your, observation which shows some,
It doesn't observations because you have seen the entire thing, because you're just seeing what it is there. You don't see in 1,000 stars, you see in 1 star. There is some problem there. So you are trying to explain.
Okay, I understand.
Yeah.
Okay, so there are no questions. Thank you. Here is Dr. Aishwarya Venkatesh from CCMB Hyderabad. So she did a PhD from University of Wisconsin Milwaukee and postdoc from Marquette University for joining CCMB. She works on the molecular mechanisms responsible for regeneration of the central nervous system. She was selected as associate in 2023. She'll be talking about decoding the molecular blueprint of regeneration in mammalian CNS neurons, peeling back the regulatory layers.
Thank you. Am I audible? Okay. Okay. Good morning, everyone. There's a lot of firsts for me today. This is my first time in Goa, first time I have family in the crowd. My first meeting as an associate, but the first time I'm going on the heels of an astrophysicist. I never thought, I've spoken to such a diverse audience. So my goal today is not to drill into the nitty-gritties of what we do, but rather use this as an opportunity to communicate the excitement for our science to a really diverse set of minds. And even if I can get a few of you excited about our problem and think along with me, I'll take that as a win. Okay, so I put one word in my title in green, hoping that's the one thing that everyone might know. Can I see a quick show of hands? Has everyone heard of the word regeneration at least once? How many of you? Let's just see how steep my challenge is. Okay, I see a couple of hands going up. So regeneration is basically the ability to replace lost cells, tissues, structures, organs, and even, you can even regenerate your entire body from scratch. It's been observed since the dawn of time. fully be back to normal. But the word regeneration was coined in 1700s by Tremblay, who was actually confused whether hydra was a plant or an animal. And at that point, the reigning theory was that if you cut something, if it grows back again, then it's likely a plant. And if it doesn't, it's an animal. And hydra posed a problem to him because he cut it. It behaved like an animal in the way it was feeding, in the way it was attaching to structures, but it continued to grow from wherever you cut the arms off or tentacles, if you will. So he coined the term regeneration to say that there are certain animals that are able to replace completely lost tissue. And then moving on to neuroscience, which is the focus of my work, this is Professor Ramoni Cajal. You might know him as the father of modern neuroscience. His intricate drawings are rivaling the confocal images that we take today. They still match up to the finest detail. So that tells you a lot about how talented he was. He noticed that this is a spinal cord, right? So he noticed that if you sever the spinal cord, instead of noticing repair like we're observing with other species, the axons, which are the cables that connect the brain, and I'll get to it in the next slide, they sort of turn away from the site of injury. They absolutely don't want to repair or grow past. So Cajal was the first person who actually coined the term nervous system regeneration to say that it's in the brain, regeneration seems impossible. It's the challenge that we're still tackling centuries later. So I want to introduce you to the spinal cord. We work on regeneration using the spinal cord as our system. And the primary cell type in the brain are neurons. They stay up in the cortex, they never move. So if the cell bodies are right here, they send out these thin cables called axons, and a bundle of such axons pulled together forms your spinal cord. And what happens is the spinal cord is activity center, pretty much controlling everything you do, from walking, sensing, talking, moving your hands. All of it is controlled at different levels of the cord, right? And normally what happens is you have signals going back and forth. It's like an information superhighway ferrying constantly. And without an injury, in the absence of an injury, things can go back and forth. But what happens if there is an injury, right? Can be in the form of a trauma, it can be stroke, it can be a variety of reasons. What happens is the cells never die. They're alive. They're very much alive. They continue to send messages down, but the messages get kind of halted up at the site of injury. They never make it where they have to, right? And this is exactly what Cajal explained in his book, where he says, all the observations lead us to the conclusion that the central tracts are incapable of regeneration. And sadly for us, his harsh decree is true even today, right? So if you've seen or heard of patients with paralysis, they've all suffered an injury at some level of the cord. Depending on which level of the cord they were injured, they lose those functions everywhere below the site of injury, and they retain functions above the site of injury. And right now, as we speak, there are no treatments. There's nothing in the clinic. All you get is some rehab on how to sort of have some muscle function, and then you're sent back home, okay? So the same problem that Cajal noticed is the problem we're tackling in the lab as well. But after the depressing news, here's some positive news, right? Almost all of the species on the evolutionary spectrum, starting from lamprey all the way to mammals, in their young ages, they're fully capable of regenerating and repairing their nervous system, including us. But when you look at adults, there's a shift now. Some species continue to retain the ability for repair, and then there's a drop. In mammals, you notice that the ability for repair is only present for about a week after birth. This is how a mouse pup looks on the day of birth, on its birthday. And then a week later, you'll notice that they have started regaining some movement, and at that point, the ability for repair kind of shuts off, and it remains off in an adult mice, right? So this is sort of the fascination I've had. What is it that happens in that 1 week that takes a neuron from being able to fully repair and regrow to completely not being able to? And to sort of communicate that, I'm going to take you through a metaphor, hopefully something that's accessible to everyone, irrespective of whether you've been introduced to neuroscience or not. So here's a warehouse, and assuming that you want to build a way to connect this warehouse to all of the houses and some endpoints, what you would normally do is recruit a set of engineers who have a blueprint saying, okay, so this is the problem we have. We want to connect this place with all of these places. These guys come up with a blueprint. They read the blueprint, and then they recruit a bunch of workers that are going to sort of do the heavy work of building the road from scratch in chunks, right? Now, the key players in this process are the engineers and the workers. So what happens if there is an injury in the road at this point, right? Let's say there's a damage. everyone's already on site. They know what to do, they know how to fix this, so the repair is swift, almost back to normal. But what happens after the construction is completed, right? So everyone is gone. They don't seem to want to go. Everyone is gone, and at this point, if you want to bring everyone back to the same spot, when cars are starting to ferry, then the problem is a little bit more tricky. Now you have to remember who those original engineers were. They have to remember who the workers were. We don't even know if that same team is going to be able to fix the problem that we have. And so there's a bunch of challenges that will determine whether the road gets back to where it was, how quickly, at what extent, so on and so forth. Now, building the spinal cord is no different, okay? So we have the brain right here, and it needs to lay a thin, long road to connect to the rest of the body, all the way through the length of the cord. And the engineers in this case are these pro-growth factors called transcription factors. They're master regulatory proteins. They are the ones that are reading the blueprint. They know which genes to turn on, when to turn on, who gets turned on together, and who works together for how long, all of those. And the workers, who are in hundreds, are these genes of different functions. They are complementary functions, right? So you have things that remodel the cytoskeleton. In simple terms, it means making up the building blocks for a growing neuron. It's growing at 3 millimeters a day, which is a lot of cellular material, right? So you have genes coming together that make the material, make sure it's going to the right spot, make sure that there's no diversions along the way, that it gets synthesized here and it goes exactly where it's supposed to. And then powering the whole process is a bunch of genes that are, important for energetics. And then, of course, like I said, you have to synthesize all of these molecules. So these transcription factors sort of recruit these different groups of genes during development to build the spinal cord. And what happens is, between this age to this age, the cord is getting laid, the road is getting laid. There are no messages, there's no confusion, and everyone's on site, right? But about a week after birth, now the neuron is not growing anymore, it's grown where it has to, it started connecting to its targets, right? At that point, these people are sent off, they're turned off because they're no longer needed. And what happens is, species or neurons that are able to, upon injury, correctly bring these proteins back up, which then turn these on, successfully recover. And the ones that don't, like mammals, end up having permanent irreversible damage. So I'm going to bring back this spectrum I showed you before. So here's the regenerative ability in adults. I'm going to layer it with one more layer. Here are the list of animals that are able to reactivate those engineers back on after injury. And you'll see it's 100% overlap, right? The ones that can bring those people back to site recover, and the ones that don't, never. So then, for the bulk of my training as a graduate student, postdoc, and my own lab in the US before I moved to India, I've been chasing this idea that we know that transcription factors, the inability to bring them back to site is what causes the problem. What if you go in and artificially put in those genes that are needed, put in those transcription factors after injury? Is that sufficient for repair and recovery? To what extent, right? And we've put out, and this was a big black box. We didn't even know whether developmental factors are sufficient for repair. If you bring them back, are they going to be able to do what they do? And are there other barriers apart from transcription factors, apart from the engineers, that can cause issues? So these are sort of the kinds of things that I was working on. So the first thing I'm going to show you is, yes, if you do bring back the developmental factors, they do help in repair. And this is where there's going to be a little bit of data, so stick with me. So what we do is we prime the brain by injecting our gene treatment into the region of the motor cortex that projects to the cord. And then we allow the genes to be expressed and challenge the animals with an injury about a week later. And then what we do is we look at the cord and we decide, is there regenerative growth or not? And to look at the cord, to be able to visualize the cables that are axons, we add GFP as a tag. So what you're seeing here in red is the site of injury, and in green are the axons coming down from the brain. And I hope you'll appreciate that right at the site of injury, they just stop. There's absolutely no regrowth following the injury site, right? So if this is your cord, imagine if this is the cord, you're looking at it in this angle. You're looking at it from the side, right? You're going through the length of the cord. And here, with one of our gene treatments called KLF6, which is a very important factor for developmental growth, you'll see well past the injury site, these white arrows are pointing to the long distance growth of axons. So we do manage to reboot the growth program. And the next thing I did is if KLF6 does so well, transcription factors are known to be very social in nature. They work with interacting groups. So if we can find factors that KLF6 works with during development, can we boost the effects that we see during injury, right? So now you're looking at it from a slightly different angle. It's a different injury model. So if this is your cord, you're looking at it from the top, okay? So that's where your alignment is. So just to align you, here's the two butterflies of the cord, the halves of the butterfly. So here's your uninjured site. Every blip of green you see is an axon lit up with GFP. When you injure them, there's rarely any growth past the injury site, right? So this is your midpoint and then different points post-injury. Now, when you supply KLF6 alone, like I showed you here, it's a different injury model, but we see the same effect. We do see some axons lighting up past the injury site. But if we go in and supply a factor along with KLF6, its friend from development, now suddenly we see a doubling in regeneration, right? I think you just see a lot more green on this side, and this translates well to behavioral recovery as well. We see that with just KLF6 versus KLF6 with its partner, we see a lot more growth and behavioral recovery. And so this is sort of where I wound down my time. I summarized my entire decade of work in 2 minutes, but that's what we do. So it's promising that we can even induce regeneration because in the history of our field, we know of only 3 or 4 transcription factors that can even do this, and 2 of them came from our group. But then when you let the dust settle and the euphoria settles, when you take a reality check, we notice that in development, we get 3 millimeters of growth in a day. And here we get 3 millimeters of growth in 3 months or more, which means for a patient to actually be able to use this treatment, if they have something like a lumbar injury, which is way down, it's going to take them 25 years for them to tap into this gene therapy. So we really need to do more and we need to do it faster, right? So that's the challenge here. How can we reprogram these neurons back to the developmental levels of growth? So that from a fundamental point also it's interesting. Why can't we get it back to where it was? It's after all just the genes that have changed. And also from a translational point, how can we make this more clinically feasible? And so those are the kinds of things that, like I said, I tackled in my work, and what are the barriers? I'm going to be focusing only on transcription factor related barriers, but we've also identified there can be a lot more layers to this puzzle. And then when I opened my group in CCMB, it became clear that for us to really rebuild the cord following injury, we're going to have to identify the full set of factors involved during development. To give you a hint of what that means, it is a hairball, but just stick with me. This is basically how a regenerating neuron, when it's successfully able to repair itself, how it responds to injury. It doesn't recruit one KLF6, it doesn't recruit one NR5A2, it recruits tens and tens of factors in very discrete phases. This is immediately post-injury, few hours post-injury, few days post-injury. So we're looking at engineers at the scale of tens, not ones, right? That was the first thing. And the field where it stands now, we know of maybe 5 to 6 factors. So there's definitely a very important puzzle is who are the rest of the factors? Who else is out there coordinating growth during development, and how can we tap into them for regeneration? So this is a challenge I took up in my lab. This is my postdoc, Manoj, who's a stellar bioinformatician, and my first graduate student, Yogesh. So we started from ground zero. We tapped into a newer technology called single-cell genomics. I won't get into the specifics of that, but what it allows you to do is beautifully take neurons from different age and map them onto a trajectory as they lose regenerative ability, right? If you really want to know how to reprogram back, you know how they were programmed in the first place. That's where you start. And so here we have embryonic neurons, and then you have different ages of neurons going all the way to P28 and older. And every violet blip you see there is a junction as they're transitioning in this capacity, going from highly regenerative to no regeneration. So then we decided we'll anchor on those junctions and pull out who are the factors that are driving the transition, right? Who are the factors that are making this happen? And we found that all of them belonged to a single family of transcription factors called nuclear receptor factors. They're very, very well studied in biology, but surprisingly hardly studied in regeneration. And we started with just looking at how they're expressed across development. So here you are looking at an RNA-seq, which is an expression map across time. And what you'll notice is they have distinct patterns. Some of them come up very early. Some of them come up right on the heels of the early factors, sort of mimicking at the same kind of hierarchy we observe in biology when regeneration successfully happens. So we feel like we're on the right track here, and I'm only showing you expression data here. The another very encouraging thing was when we went back and looked at the species that can regenerate. By the way, mice can regenerate their peripheral nervous system just fine. They only have issues with their central nervous system. all of these neurons respond to injury by triggering these factors in the same wave. So that was giving us another layer of confirmation that, okay, we're headed. And then finally, the hallmark of these factors are they sit on DNA and they activate genes. So we asked whether not only are they present, but did they actually sit on the relevant DNA? And I have data for that as well. So they all seem to be lining up where we think it is. So our lab is about a year and more old in CCMB. So what we've done now is, this is Yogesh's project. Hopefully if I get a chance to talk to you guys again, I will love to update you on where this lands. But for now, what we've done is we've identified the factors, we've cloned them, and we're sort of in different stages of processing this. And so our hypothesis is, irrespective of whether development or regeneration, these NRTFs are going to be important because we have hints that they are. And for the work that we do, it's really from molecules to behavior. We're manipulating neurons with the DNA, so we clone them, and you can't take your gene and just inject it into the brain. We have to make these custom AAV vectors. So the last 1 year has been about assembling the technical toolkit that we need to sort of pull this kind of project off. And so now we can, in India, I think very few labs are making their custom AAVs, but we're happy to say, thanks to Yogesh's perseverance, that we can make AAVs at least 2 or 3 a week. And they cost $2,500 a pop if you have to buy it. So that should tell you what kind of a bottleneck we've removed. And then we have surgeons who inject them into the brain right there in the motor cortex, and you can see the neurons lit up in GFP and the axons that are projecting. So that sort of sets the stage for the gene delivery. And then, lastly, we have a spinal cord injury model. You see the mice fixed on the frame, and all you need to know is this is a portion of the cord that looks a little thinner. That's where we've done the crush. And instantly, there's paralysis of the hind limbs, right? And post-injury, what we do is we allow the mice to run on, let's see if I can play that. I was going to show you a nice video of mice running on a treadmill, but it doesn't seem to be working. So we mark these different points of the mice as it's running post-gene treatment. It's a machine learning algorithm that will tell us how the mice is moving. Is it going back to its original phenotype? Where in the pipeline are we in repairing the mice to full recovery? And with that, I want to bring you back to Square One, where we started. Hopefully, there's a lot more green now. I've told you a little bit about what we do in the lab in trying to decode the blueprint. Who are the engineers? Who are the workers? And how can we bring them back to the site after injury to mount a full response? It takes a village to raise a child and multiple villages to raise a lab. I can tell you that from experience. This is my lab, a huge shout out. These guys do all of the hard work. I do the fun part of coming and talking to you about it. And CCMB has been terrific for a new faculty to start off. We have support systems that are incredible. So I want to give a huge shout out to all the people, and of course the funding sources. And last but not the least, the Academy for giving me a chance to talk to you all about this work. I think I finished a minute ahead of time. Thank you.
Thanks for giving us one extra minute. So we can take few questions.
Very nice talk.
Thank you.
Two, three things. Number one, in the humans, the Human Cell Atlas project has already generated a lot of data, both in terms of spatial transcriptomics at the single cell level as also, and we see that there is, across individuals, there is a huge amount of variation,
Absolutely.
both spatially and, temporally. Do you find that if you, cut off or make inflammation, accidental cutting off of neurons, is there a lot of inter-individual variability even in the mice in terms of regeneration?
Absolutely.
And there is. So then there is a lot of scope for understanding the spatial organization of these cells and temporal organization of the transcripts.
Yeah, absolutely. So the first thing I should tell you is, so we are tapping into single cell genomics now as well. So until we moved back to CCMB, we used to do bulk RNA-seq. The resolution we were getting is nowhere close to what we can get now, right? So we can actually go back when you do the injury, because it is surgical by hand, there's inbuilt variability, which sort of lends itself well to ask this kind of question. So even if the surgeon wants to be precise, there's always going to be one that gets crushed a little bit more, right? And you'll see that directly being reflected in the transcript level. So you'll see that the stress response genes and all of them get turned off. So instead of thinking about it as a problem to fix the variability, we're actually tapping into it for insights. And I think you're absolutely right. We should be able to use that, and that's kind of where we're hoping to be headed as well.
Hello. Nice presentation.
Thank you.
So, if you see the first half of your presentation, you show that one factor, KLF factor, will regenerate the brain part. In second part, you're saying lot of factors are required. So I'm surprising how one factor introduction in that injuries recover at least some of neurons. So what is the reason for that?
Yeah, so first of all, it's a blip usually, right? Without any gene treatment, if you cut the spinal cord, you will get nothing. Your question to why KLF6 does something at all is just to speak to the level of hierarchy that is there in the system, right? So there are going to be some factors that are higher up in the cascade that when you put back in, they are able to then trigger all of the downstream cascades. So the reason we see any blip at all is because KLF6 is a master regulator that is able to trigger some portion of the network. Now, why do we need lots of factors? That, like I said, the rates of regeneration we see is nowhere close to the developmental scale. So if you put KLF6 and if you're willing to tell a patient to wait 25 years to recover, fantastic, we are done. We can close shop today. But that's not where we are, right? We really want to be able to go back to reprogramming the neuron to what it is capable of in a younger state. And for that, you need the whole gamut of factors. So the fact that KLF6 works is like a sanity check. It's the proof of concept we need to know to sort of invest in trying to understand how to figure out who else is involved in the system.
So I have a question here, front. Good talk. With KLF6 is the Krupel-like factor, which is very connected with the cancers.
Absolutely.
So now what you are saying that it actually allows the neuronal, these cells to proliferate to some extent. So is there any connectivity that it is directly linked to the cell proliferation, and that's why you are getting some regeneration? But how much it can do for the real case scenario?
Right.
You can be the best person to
Yeah.
answer.
So yes, scale-up is involved. So one of the things that is striking to us always is many of the genes that we work with, because of how similar cancer is to axon growth. In both cases, there's bulk production of material. There is trafficking of that material to the right spot, which is guidance. You'll be surprised that there's a lot of interplay. Now, the immediate follow-up question is then how can this be looked at from the lens of gene therapy? Like you don't want to be injecting an oncogene and having that person recover from injury, but hey, look, now you have cancer. That just doesn't fit. So what we do when we do our experiments is when we inject it, we have a molecular readout, right? Always we have a list of what genes are expressed, when they're expressed, and rarely do we see any cancer genes pop up at the transcriptomics end. And I think this is, and that's a whole separate conversation we can have about how the brain has protected itself from cancer. As you know, it's one of the few organs that is highly resistant. So there is a lot of overlap in the kinds of things they're doing, and that is why I think the factors are involved, but we rarely see any sort of non-specific or off-target effects from these oncogenes. So whatever KLF is doing in that context is not mirrored at the transcriptome level in our problem, although they do similar things, if that makes sense.
You can link it with the glioblastoma.
Sorry?
Glioblastoma, that kind of.
Oh, yeah. I mean, yes, absolutely.
No, great talk, Shurya.
Thank you.
So let's say if you are saying this KLF is involved in regeneration, I'm assuming that it's the same thing happening in other evolutionary models.
Absolutely.
So have you guys seen, or maybe someone else has seen, that if you mutate those in, let's say, starfish or some other lower eukaryotes, does it
Yeah.
stop the regeneration?
Right, so fascinating question. So I actually started with zebrafish for my PhD. I actually pulled out KLF6 from my time as a PhD student, noticing that in the zebrafish optic nerve, when you crush, KLF6 is one of the first factors to show up. So then I moved it to the mammalian system. Now, how far KLF, so you're asking me whether KLF6 is known below zebrafish also? Is that
Yeah,
lower
maybe
on the...
zebrafish or maybe lower, but will mutating it or will removing it will stop the regeneration?
Shorten the question.
Yeah.
If it is going to a discussion, we should do it.
I see. You're asking about knockdown. You want to know if you take down KLF6. Got it. Yeah, if you take down KLF6 in the zebrafish model, there's a reduction, but it's not gone completely. That tells you about the redundancy of the other factors.
One last question.
So that was an excellent talk.
Thank you.
So you talked about the obvious disadvantages of not having the capability to regenerate in higher organism. My question is, are there any advantages of not having the ability to regenerate in higher organisms?
Yeah.
Right, like mammals?
Absolutely. Evolution is clever, right? I mean, the reason why it's taken so much effort to turn it off at multiple levels is because I think the complexity of the nervous system and what it would take to coordinate successful regeneration to the level of recovery. So if you have a few axons, now think about it, they're coming back into a grid that is completely miswired. So you'll have to plug it into multiple things to figure out what is going to fit. And that multiple thing might actually be detrimental to the animal as well. So this is one project we have on regeneration, but we're simultaneously looking at how to integrate that to make sure that the axons that are growing are actually growing back where they should. So I think the reason why it probably got shut off is the level of complexity that exists in the higher organisms. And you'll see this with one of our gene treatments called SOX11. The axons were growing robustly, but the animal was just doing backflips. It was not doing what it was supposed to do. I think it was that kind of, that's probably what the reason I can think of for why it was shut off in the first place.
That makes sense. Thank you.
Thank you.
So before I hand over the mic to the organizers, I want to thank you as well as the other two speakers for the excellent talks. And especially we started off with the male-female brain function difference. We went to stars, looked at the anomaly in the lithium anomaly in the red giants, and came back to again the brain function related issue. I must say that the central nervous systems. Yeah. So thanks again.
Thank
to
you.
all the
Thank you,
speakers.
everyone.
That was lovely. Thank you. And we have tea outside. And please come back by 10:45 for the next session. can hear the bell ringing furiously and frantically to get everybody in. So, welcome back after the tea break. It's working. Okay. And I invite Professor Chadda to chair this session and to be as tough as possible about time.
Blame it all on you.
Oh.
So in this session, we have one lecture by a fellow and two lectures by associates. The first lecture is by Professor Namboothri. He is with IIT Bombay. He did his PhD from Indian Institute of Science and then postdoc at Bar Ilan, Israel, and a couple of universities in US, and came back to IIT and then joined IIT Bombay, where he has been for almost 25 years now. So I invite Professor Namboothri to please come and give us his lecture. And as Madam just said,
yes, yes, thank you. Thank you. So thank you, Professor Chadda, for your kind introduction. And good morning, everyone. At the outset, I would also like to thank the Indian Academy of Sciences, Bangalore. And I would also like to thank the Indian Academy of Sciences, Bangalore, and BITS Planning for giving me this opportunity. So the title of my talk today has to do with synthesis and applications of certain polycyclic cage compounds as high energy density materials. So if you go back to history more than 2000 years ago, Plato has predicted that there are only 5 geometric figures that can have identical convex faces, and those are tetrahedron, cube, dodecahedron, octahedron, and icosahedron. Now, only 3 of them can have hydrocarbon analogs due to valency restrictions: tetrahedron, cubane, and dodecahedron. These geometric figures have remained an academic curiosity until recently. Only in the last couple of decades, cubane has been projected as a high energy density material, and it has also been used as part of a chiral ligand and also attached to medicinally active compounds. And this octanitrocubane is considered to be is a highly energetic explosive. Now, we have had interest in the chemistry of polycyclic cage compounds, particularly the bis homocubanes, with 2 additional carbons compared to cubane. And we have investigated an equilibrium between cage vinylidene carbene and its corresponding acetylene, and found that the carbene is both kinetically and thermodynamically more stable than acetylene, thanks to the enormous strain that is present in the molecule. Unlike the open chain systems where the carbene spontaneously rearranges to the corresponding acetylene. Now, I'll focus today on the energetic properties of cage compounds. As you know, energetic materials are used both civilian and defense applications. Although we have carried out the synthesis, many of the properties were evaluated by my collaborators, the combustion engineers, Professor Arindrajit Chaudhary and Neeraj Kumbhakarna from Mechanical Engineering Department. Again, if you go back to history, this is chronology of energetic materials. China has used black powder for the first time, and then Nobel developed dynamite, and TNT has been used as first generation energetic material, and RDX and HMX are extensively used. And these are the 3rd generation nitro compounds like Cl20, octanitrocubane, and also hydrozinium nitroformate and ammonium dinitromide. But none of these have been scaled up for practical applications. So what are the basic requirements for a compound to exhibit energetic properties? As far as propellants are concerned, we look at the specific impulse. Specific impulse is basically the the thrust or the impulse delivered per unit propellant consumed. So it is F by m, and it is expressed in seconds. Now, if you multiply specific impulse with the specific gravity of the mixture, then it is a density specific impulse, rho specific impulse, which is particularly important for high dense materials like the cage compounds. And if you take the standard RP1 rocket propellant fuel, the ISP is 366.2, but rho ISP is slightly higher, and as a liquid propellant. And if you consider solid propellants like ammonium perchlorate, HTPB mixture, where HTPB is binder, ammonium perchlorate is the oxidizer, the ISP is 275, whereas rho ISP is 438. Now, other properties are important as far as, for explosives like oxygen balance and detonation pressure, detonation velocity, burn rate, heat of formation, ignition characteristics, etc., and various thermal and mechanical properties. So initially, we have compared the properties of conventional hydrocarbons with cage hydrocarbons. As you can see here, conventional hydrocarbons have negative heat of formation, low density, no strain, whereas cage compounds like cubane have high positive heat of formation, high density, and high strain energy. In spite of that, such compounds do not undergo decomposition. Therefore, cubane-like compounds are considered as kinetic rocks, but thermodynamic powerhouses. And where does the energy come from? The energy comes from the angle strain. As you can see here, cyclopropane has the highest angle strain, and the heat of combustion per methylene is highest for cyclopropane. So if you have cubane-like compounds with several cyclobutane rings fused, in addition to angle strain, there is torsional strain as well, which is stored in the structure of the molecule. So these are some of the properties of cubane: high melting, high boiling, low decomposition, high density, high vapor pressure, high positive heat of formation, enthalpy of sublimations, strain energy, etc. So, these are the properties expected of such cage hydrocarbons: faster ignition, higher heat capacity, superior burn rate, greater heat release, at the same time kinetic stability compared to conventional fuels, and these do not require any cryogenic conditions for storage, and these are also less sensitive. So initially we have calculated the heats of formation and specific impulse of cubane and several other hydrocarbon materials. As you can see that cubane has the highest, all of them have high positive heat of formation. Cubane has the highest specific impulse, followed by methylcubane, and then other cage hydrocarbons also have high specific impulse. And we also notice that if you take the dimer has higher specific impulse compared to monomer. For example, this bis-homocubane has an ISP of 365 seconds, whereas its dimer has 371. So there is a 6 seconds improvement in terms of specific impulse. So considering that, we have synthesized this bis-homocubane dimer in 4 steps, starting from readily available dicyclopentadiene in excellent overall yield, and found that these have, this has high positive heat of formation and also ISP much better than that of conventional hydrocarbon fuel, the rocket propellant fuel, RP1. At this point, the VSSC scientists have advised us to introduce energetic functional groups like nitro, azido, and so on to such cage hydrocarbons, because there was some precedence in the literature. Tris-homocubane, the tetranitro tris-homocubane was considered as, was less shock sensitive than TNT. At the same time, it has an energy output comparable to that of an RDX TNT mixture. Similarly, octanitrocubane is more powerful than HMX. But synthesis of these compounds on a large scale for practical applications is a challenging task. Therefore, initially we focused on bis-homocubane skeleton with 2 additional carbons compared to cubane, which can be prepared in a couple of steps, starting from readily available dicyclopentadiene. So this cage diol was synthesized, which was then nitrated to get this dinitrate. And as you can see here, the dinitrate has a rho ISP of about more than 45 seconds higher than that of RP1. And we have also converted this bishomocubenon to several nitro derivatives, both unsaturated and saturated, and also the one containing two nitro groups. And in all the cases, you can see the specific impulse of the RP1. So here these compounds are added to RP1 about 30%. That improves the specific impulse by about 20 to 76 seconds. And if you add these compounds to a solid propellant combination like AP, ammonium perchlorate, HTPB, or replace HTPB with these compounds, then also the specific impulse is much higher, about 30 to 25 to 30 second improvement. We have again prepared this cage diol in good quantity and converted it to this bis azide, which was then converted to this bis triazole and also this bis tetrazole. And you can see that addition of 30% of any of these compounds to RP1 would improve the specific impulse by about 14 to 83 seconds. And also replacing HTPB in the solid propellant combination would also improve the ISP by about 20 to 35 seconds. And even though there are more number of steps involved in the synthesis of homocubane, that is one carbon homologue of cubane, we synthesized this homocubane carboxylic acid in about 6 steps and then looked at its ISP. So as a liquid propellant, this would perform much better than RP1, more than 19 seconds improvement. And as a solid propellant additive, also this would perform with very high ISP. And we have also synthesized several other derivatives, which are, of course, not that energetic. And we converted this homocubane carboxylic acid to several derivatives like this methyl homocubane, hydroxymethyl cyano tetrazole derivative, and also oxadiazole. So in all the cases, these compounds performed well as a liquid propellant additive, 30% to RP1, and also as a substitute for hydroxy terminated polybutadiene, that is HTPB. And we have also converted the homocubane carboxylic acid to its ester and another oxadiazole and this hydrazide and also this amino oxadiazole. In all cases, the ISPs are much superior compared to RP1 and in liquid propellant combination and also compared to HTPB in the solid propellant combination. Now, we also converted the hydroxymethyl homocubane to this triazine and also this triazole, amino triazole containing 2 homocubane moieties. And in both cases, you can see the, these 2 compounds perform much better compared to conventional liquid propellant RP1 and solid propellant binder HTPB. And so at this point, we thought that even though it takes several steps to synthesize cubane, it takes about 8 steps to make this cubane dicarboxylate. We prepared it, and we thought we would evaluate the cubane derivatives as well. And we have synthesized this tetrazole, monotetrazole, and also this bis oxadiazole. And in both cases, as a liquid propellant and also as a solid propellant, these compounds performed much better. As liquid propellant, more than 27 to 33 seconds improvement, and also as a solid propellant, very high, more than 100 to 140 seconds improvement compared to HTPB. Finally, we went back to bis-homocubane because it's easier to synthesize in a couple of steps from dicyclopentadiene. So we prepared this cage keto alcohol, which was converted to this dinitrate and also this ketonitrate, dinitroalcohol, azide, and also this trinitro compound, dinitronitrate. And as highlighted, you can see that these compounds perform well as liquid propellant additive with about 20 to 30 seconds improvement, and also as solid propellant, substitute for HTPB in solid propellants. The cage keto alcohol was also converted to this keto azide, and by accident we have got this cage dimer when we attempted to reduce the azide group, and this amide was also isolated as a side product, and all these compounds have shown much higher specific impulse, both as a liquid propellant additive and also as a substitute for HTPB in solid propellant. So these are the compounds we have synthesized and evaluated, and these are the bis-homocubane derivatives, and here we have the homocubane derivatives, and couple of cubane derivatives also have been synthesized, and evaluated, and their performance in terms of ISPs are shown here. And so as I told you in the beginning, we have carried out synthesis and characterization of these compounds. The thermal analysis, calculations, all the decomposition pathways were all carried out by our collaborator in the mechanical engineering department. For instance, the thermal decomposition was investigated by using TGA, and the gaseous products were analyzed by rapid scan, FTIR, and mass. And the decomposition pathways were evaluated by DFT ab initio, and the compounds were also, the structures were also optimized using DFT. And the propulsive properties, especially the ISPs, were calculated using a software called NASA CEA. And these properties are not important for our compounds because our compounds are not explosives. But the challenges involved are the scale up. Still, there are many steps involved to synthesize these compounds in reasonable scale, and the number of, we try to minimize the number of steps as much as possible. Also, since these are hydrocarbon derivatives, soot formation is a problem which has to be addressed. And again, our collaborators have evaluated representative compounds, for example, this dinitro compound, and found that addition of this dinitro methyl bis-homocubane to RP1 enhances the burn rate by promoting micro explosions. And also addition of this bis-tetrasol, bis-homocubane derived bis-tetrasol to AP HTP mixture, only 5%, addition of 5% is enough to improve the burn rate by about 15 to 20%. The red dots are for the cage added mixture, and the black squares are for the conventional AP HTPB mixture. So our we are trying to improve the performance of conventional fuels, liquid propellants like RP1, and also the solid propellant AP HTPB mixture by adding our cage compounds as additives. So this is one of our interests, the application of polycyclic cage compounds as energetic materials. We also have interest in other, for instance, development of new synthetic methodologies, mechanistic studies, and synthesis of other materials with interesting properties. And quickly, some of our properties are, some of our projects involve alpha functionalization of nitroalkenes under the Morita-Baylis-Hillman and Rauhut carrier conditions, synthesis of polynitrogen containing heterocycles using diazo compounds, especially Bestman or HERA reagent, catalytic asymmetric conjugate addition of active methylene and methine compounds to electron deficient alkenes, synthetic applications of curcumin, the yellow pigment that is present in turmeric, in cascade reactions, magnesium, application of magnesium bromoform reagent system for introducing tribromomethyl and also dibromomethylinated groups in various electron deficient alkenes, and then application of this sulfonyl phthalate as a 1,4 dipolar synthon for the synthesis of various fused and spiro carbocycles and heterocycles. Then in collaboration with a French group, we are working on application of carbon nanotube metal nanohybrids as heterogeneous catalysts in various organic reactions. We also have interest in interest in various Michael-initiated ring closing reactions. So yeah, this is my group, the latest picture, this is the last year's picture. And the students who contributed to this program on energetic materials are here. Bichismita Sahu, woman. So Bichismita is at NIPER, Ahmedabad. Woman is a faculty in Finland. And Narasimham is in a company in Hyderabad. Guru Raja is faculty at Central University, Gujarat. Sohan is a postdoc in the US, and she is still continuing her PhD, Tejasri. So I must thank all my students who contributed to this project, my collaborators, especially Professors Chowdhury and Kumpagarna, and also Sudarshan Kumar from the aerospace engineering, and their students. Some of them got Best Thesis Award for work in this area. And collaborators, Dr. Nainan, who retired long ago from ESSC, Dr. Verghese and Dr. Reshmi, and collaborators from DRDO, Dr. Arvind Kumar, Nandagopal, and Rohit, and most of the facilities we have in our IIT. And I must thank the funding agencies, ISRO and DRDO, for their support. Thank you very much for your attention and patience.
Thank you, Professor Namboothri. You really did an excellent job, both of giving us a glimpse of the vast landscape over which you have done your research and of sticking to the time. We have time for a few questions. Yes, please. Swapan.
Very nice talk.
Thank you.
Now these energetic materials are thermodynamically very unstable, but must be kinetically stable, right?
Yeah.
So have you looked at the activation energy barrier or something like that?
Yes, we have calculated the bond dissociation energies of those cage compounds. And we found that the strained rings break first, especially the cyclobutane ring. We found that. Yeah. So we have evaluated the decomposition pathways. I mean, since the time is short, I could not present any of that. And some of the functional groups break first, like nitro. That also we have observed. Thank you.
Yes, please. The mic is coming. Just a minute.
How do you address the concept of decomposition pathways?
Yeah.
Because that's something which is very important for all these compounds.
Yes, so that is why we have the hydrocarbons as such do not undergo decomposition that easily. So we have introduced the energetic groups like nitro, azido, etc. And we found that the energetic groups first break away from the cage, and if it's homolytic decomposition, a radical would be generated, and then that would trigger the decomposition of the cage. Especially the highly strained rings break first. So there is an activation energy for decomposition.
The concept which I was trying to understand was, is there always a unique pathway for that?
A unique pathway? No.
It
It changes depending upon the functional groups attached to those compounds. Yes. So each compound we evaluate.
I have a layman's question.
Yeah.
You were talking of both liquid propellant and solid propellant based
Right.
on the same cage compounds.
Yeah.
Do
Yeah.
you have any general comment on liquid versus solid?
Yes, some of the compounds are solids and some others are liquids. But the liquids are highly viscous liquids. So we could add that to solid propellant combination as well, like APH TPB.
Physical properties are quite similar.
Yeah, quite similar. Yeah. These are not free-flowing liquids like the RP1. RP1 is like kerosene, refined kerosene or jet propulsion fuel.
Okay, if there are no further questions, we return our thanks to
Thank
Professor
you.
Namboothri.
Thank you very much.
Next speaker is Professor Haripada Saud. He's currently at ISR Pune. He holds a PhD from Institute of Science. He has done postdoc at IIT Bombay, Virginia Tech, IIT Guwahati, and at TIFR Center for Applied Maths. So he was selected as an associate during the last year. We look forward to hearing you.
Hello? Yeah. No, I don't have. First of all, I would like to thank the Academy for giving me this recognition and for the opportunity to speak here, and the organizers for organizing this meeting so efficiently. I work in Hilbert space operator theory. So what that means is, so I will explain what Hilbert, you know, most of you. So Hilbert space, you have a vector space and you have equipped with an inner product, and with respect to the induced norm, it gives you a metric and the vector space is complete. So it's a metric space. Metric comes from norm and the norm comes from inner product. So there are so many structures into it. You can think of it as just Cn, okay, n-dimensional complex Hilbert space. And operators are linear transformations between two Hilbert spaces, preserved structures, linear structures, which you can think of as matrices, okay? So this is a joint work with my first postdoc mentor, Bhattokrishna Das from IIT Bombay. And the problem that we proposed could not solve, only partially we solved. So the purpose of this talk is to just tell you what this problem is about. And okay, so take a compact set, a closed bounded set in the d-dimensional complex space. Please, if you want, you can think of it as just the complex plane and a tuple of commuting operators. So it's a d tuple of commuting operators. If d is equal to 1, then this is just a single operator, meaning linear transformation. You can think of it as a matrix acting on a Hilbert space H. You say that K is a spectral set, okay, for T. If for every polynomial f in d variables, if d is equal to 1, it's one single variable polynomial. The norm of ft, so you plug in t in place of z, so if it's a polynomial, and you look at the norm, operator norm, and this is what that means. You're looking at the supremum of fth, the norm of fth, where h runs over the sphere, and that should be less or equal to supremum of f over k, the compact set k. If that happens for every polynomial, then you say that it's a spectral set for T. You say it's a complete spectral set. If this happens, not only for scalar valued polynomials, but also for n by n matrix valued polynomials, okay, for every n greater or equal to 1. So, the one easy way to think of a polynomial with matrix valued is it's a matrix with polynomial entries, and you know what? So f of f is equal to f of z equal to f of f sub ij z, ij equal to 1 to n, and so f of t is equal to f sub I z t. So you look at the operator norm of that, and that should be less or equal to the norm of f over the compact set. So from the definition, you see that a complete spectral set in particular is a spectral set because complete spectral set is asking more. You want that to happen for every n equal to 1, every n. And this is a operator algebraic way formulation of the problem. This is operator algebraic terminology, and this is an operator theoretic way of looking at it. Okay, so this happens equivalently, something happens. This I don't want to talk about today. I'm going to avoid that. This is operator theoretic way of looking at it. So the question is, so of course a complete spectral set is a spectral set. The question is the converse. When is the converse true? When is a spectral set a complete spectral set? That is indeed the holy grail in the spectral set theory, which is called the dilation problem. The dilation problem asks you when a spectral set is a complete spectral set. So you have a compact set, you are asking, is a spectral, if you have an operator, so K is a compact subset of the complex plane, T is an operator for which K is a spectral set. Is it necessary that K is a complete spectral set? So when is a spectral set a complete spectral set? Referred to as the dilation problem. This problem turns out to have a wide range. So it's yet not, there is no characterization of such compact sets known for which a definitive answer can be given. Okay, so but the planar case, that is the case when K is a subset of the complex plane, is better understood. Okay, so I'm going to walk you through the history of successes and failures. It turns out that the geometry of the compact set comes in. Okay, in fact, we use a lot of complex analysis. So when is a spectral set a complete spectral set? The answer is when K is disclosure. So disk is also Z in the C. So the modulus of Z is less or equal to 1. So it's just a unit disk. Or more generally, when K has a connected complement. So you look at the complement of K and you give me any 2 points, I should be able to connect those 2 points by a curve. So when this happens, and when whenever this K is a spectral set, it's a complete spectral set. When K is an annulus, so there's a big hole, you have a disk and you remove a tiny disk from it. When K is an annulus, this is it's a complete spectral set, but not when interior of K has 2 or more holes. So if you have a if you have a disk and if you remove 2 disjoint disks from it, then it is not going to be a complete spectral set. There is an example. And this is also, I have cited the work, the legends that who have done this, the stalwarts. When K is a bidisk, then also it's a spectral set. Whenever it's a spectral set, it's a complete spectral set, but not when K is a tridisk. So you see, you see what tridisk, this is tridisk, a Cartesian product of the disk 3 times. So you see when K is a disk, it's affirmative. When K is the bidisk, it's affirmative, but it fails for the tridisk onwards. So it has a rather mysterious history of successes and failures. Our interest lies in the case when K is an algebraic variety intersected with the closed bidisk. And so before I propose the problem, I want to walk you through why you might want to care about dilation theory. So it gives you a very different approach to, much easier approach, I would say, to the peak interpolation problem, which asks you, so you have been given Zj and Wj from the bidisk. So Zj are distinct, z equal to 1 to n, Wj may be equal. The problem asks you, when does there exist an analytic function such that it maps the to the final data Wj. So for an analytic function, you can think of it as a polynomial but of infinite degree. Okay, you can think of it as that, and with some sort of convergence going on there. So it gives you an easy, quick proof of that interpolation problem. It gives a different approach to still open problem, which is referred to as the invariant subspace problem. It doesn't help much, but nevertheless it gives you a different way of looking at the problem. So the problem asks you, does every Hilbert space operator have an invariant subspace? Very easy to formulate, but it's not yet known. It's not really, so it's solved in some cases, like if you have a Hilbert space of which is not separable, so it's a metric space, you know what separability means. So if it is not separable, then the answer is affirmative. Every operator has a Hilbert space, invariant subspace. If you have a Banach space, then it is negative. So if you remove the inner product structure, then it fails. The problem is still open when the Hilbert space is separable. Okay, unfortunately, but there are recent 2 papers posted on arXiv this year claiming to have solved this problem. It's only a matter of time to know if it is correct or not. 2 distinct papers, by the way. So dilation theory also gives you an operator theoretic representation formula for bounded analytic functions. I don't want to go into that. So it gives you a operator theoretic representation. You have a bounded analytic function, you can represent it. You can represent it in terms of operators, which is very nice, very helpful. And it brings down the study of abstract operators acting on abstract Hilbert spaces to the study of analytic functions. The theory of analytic functions is very rich. So it helps to think of operators as functions in some way. And a concrete example to give you that demonstrates this statement is two operators are unitarily equivalent if and only if some functions, analytic functions corresponding to them coincide. Okay, so there is a notion of coincidence here. And I told you that I want to investigate the dilation problem when K is an algebraic variety. So this is what the variety that I was talking about. A distinguished variety is an affine variety. So xi is a polynomial in two variables that satisfies this. It should intersect the open bidisk, okay, and it should exit the domain through the torus. Okay, it should not intersect the other part of the boundary. For example, you can take z1 minus z2, the simplest one you can think of, or z1 square minus z2q. Okay, and for a non-example, you can think of, so you can perturb the first example by something by multiplying something that is not allowed. So you can so if for example you say, well, the open bidisk, but it doesn't satisfy the boundary condition. Because for example, 1, 0 is in the 0 set, which is not in the torus. Neither is it in the open bidisk. Well, these varieties are important. So the real importance of these varieties come up in work of Agler and McCarthy in 2000. They prove this. Okay, so very simple result. If you have T1, T2, a commuting contractive matrices. So by contractive, I mean the operator norm should be less or equal to 1. It contracts the ball. Okay, commuting contractive matrices with all their eigenvalues in the disk. So they are not allowed to have eigenvalues on the circle. You can still find a way, but in their theorem, hypothesis was written in this way. Then if you have such a pair of commuting matrices, then there exists a distinguished variety. You have seen what that means. Certain special variety such that 0 set of eta intersected with the closed bidisk is a complete spectral set, meaning this inequality is happening for every n by n matrix valued polynomial. Now, why is this important? This is very nice because such a thing was known due to Andrew when you took the supremum over the closed bidisk. Okay, please remember this is a very thin subset of the bidisk. Okay, so this is a major sharpening of this inequality. You will see that much of the main object of our work is to know whether or not the matrices are of any special kind here. Okay, we want to know if we could have this inequality happening over a distinguished variety, not over the whole bidisk, for pair of commuting operators other than matrices. Okay, we want to drop the finite dimensionality, and you will see that it could not get away from finite dimension. You will see in some way we did extend, but had to say that we could not solve it in the real infinite dimensional case. You will see. First question is, are distinguished varieties complete spectral set? Answer is no, and this is there are example. If to take this example, we saw that this is a distinguished variety. Then there is a commuting contractive matrix pair which has this as a spectral set but not a spectral set. That's what it means by it fails. That's what it means by certain set is not complete spectral set. It is a spectral set for some pair, but it's not a complete spectral set. And please note that the example is found in the finite dimension case. We are asking a revised question. We're revising the question. Okay, so this pair does not have this guy as a spectral set. But we will be happy, as far as our goal is concerned, we will be happy if this guy had a different variety as a complete spectral set. So the question is, does there not exist another distinguished variety, 0 set of eta, maybe not this, such that 0 set of eta intersected with the closed bidisk is a complete spectral set? And you know what that means here. Turns out that this is going to be a necessary condition. Okay, right. So so this is a necessary condition. The main result that we have proved is this. Okay, the extent that we could solve the problem to. So if you have a commuting contractive pair of operators, not just matrices, but for operators, it's allowed to act on infinite dimensions such that it is annihilated by something by a polynomial defining a distinguished variety. So, and we saw in the previous slide that this is a necessary condition. You could not get away with it. So let it be there. And something that you are not proud of is this. You took T1, we take T1 and T2 such that you look at this operator. So this is an adjoint. So you have a matrix Aij and the adjoint is Aj bar. So it's the adjoint of the operator T2, operators Tj. You look at that operator, look at its range, which is a subspace, look at its dimension. If that is finite dimensional, so you see, that's what I meant by we could not get away with finite dimensionality. We need some finite dimensional condition. If that happens, then there exists an eta defining a distinguished variety such that this happens. Okay, so we have been able to extend the result to infinite dimension case, but not really. I mean, we did, but this is a very stringent condition, I would say. Okay. And this is, like I said, this is an ongoing work, and we hope to, there are evidence, there are reasons why we believe that we would be able to drop this. And as you would agree that this extends earlier result of Aguilar and McCarthy, and this is not the only work that which puts an effort to extend this result of Aguilar and McCarthy to infinite dimension. My collaborator and his collaborator Das and Sarkar, they have tried this, and they had the same inequality, but they had even stronger condition there. Okay, that's it. Yeah, this is the paper where I proposed the problem, and the referee calls it a constrained anodization problem, which you like calling the problem, and this is the work that we are preparing now. Yeah, thank you.
Questions from the audience, please?
Hello. Yes. I think this belongs to the domain of algebraic geometry.
Yes, it does, and we know very less about that. Yeah.
Oh, you have talked of matrices on the one hand and analytic function on the other hand. And you are talking of Hilbert spaces. So Hilbert spaces, one is on a function space, and the other one is algebraic entity. So I just do not get how do you bring these two together on the one hand, and secondly, can your matrices be infinite dimensional?
So when you say can the matrices be infinite dimension, what exactly do you mean? Absolutely. So when you talk about operators, it's allowed to be. So you can think of an operator as a, infinite by infinite size matrix. Okay. You can think of matrices operators as matrices of infinite size. First question is, how do you relate to operators and varieties? This is because, this is again by Aguilar and McCarthy. They noticed that if you have so this is the operator theoretic side of it, okay, which I avoided for this talk. If you have a T1 and T2, then you can dilate it to V1, V2. V1, V2 are nice. V1, V2 are commuting isometries, and it turns out that this thing happens if and only if you are able to dilate this T1 and T2, whatever that means. I didn't define that. If and only if you are able to T1 and T2 to V1 and V2, which lives in such a thing, meaning its joint spectrum. Okay, the joint spectrum of V1, V2 is an algebraic variety. Okay, so indeed that is the reason why we need this finite dimensionality. We need a lift. We need to dilate T1, T2 to V1, V2, which lives, meaning the joint spectrum should be a distinguished variety. That's why we're crucially using the finite dimensionality assumption. I do not quite understand. I hope, I thought I would answer that question, but could you please repeat that? What do you mean by convergence? Yeah, sorry. Maybe we can discuss.
Thanks, Haripada. My first, I think that answer, that spectral set definition is a subset of CN, and this operator, that relation maybe if you tell that will answer the things. But my second question is, in between told invariant subspace problem.
Right.
But what is the relation with your work?
It gives you, the dilation theory gives you a different approach, different way of looking at the
because
problem.
it's a famous problem solved this year. Yeah, so is it?
Right. So the problem is still unsolved. But
no, it's solved, no?
No, I said that it's been put on archive, right?
I thought it is solved, yeah.
Right, so yeah. It just gives you a different way of looking at that.
Well, please pardon me if I ask some out of the way question.
Please go ahead.
I'm not familiar with this field of work.
Right.
But the so-called dilation appears in a fundamental way in wavelet transforms. What has it got to do with this type of result? What I mean to find out is, does your result have any relevance to the wavelet transforms which are being sort of, which have been used in signal processing and the likes as versatile tools? I want to know whether there is a relation.
I do not know, but the interpolation problem that I talked about.
Yes, it is related.
Yeah, right. That has application in control theory and circuit theory, but
no,
I don't know.
But the concept of dilation is nothing different, if I understand correctly, nothing different from some sort of independent variable scaling, fx and fax, and then you come out with the sort of shifting and all that to generate the wavelet transforms. So I was wondering whether that type of operation has anything to do with your
I do not know. I do not know, unfortunately.
Thank you.
Thank you. Yeah, we can talk about it later.
We now go to the last topic. So Vikash Kumar, CPOR work. He had done his graduate school from ISA Kolkata, and after that he has been with NCPOR for 7 or 8 years. He has been selected as a I mean now he needs to talk. I'm sorry.
Thank you, sir, for the introduction, and I would also like to thank the Academy for this opportunity. The title of my talk is Polar Climate Reconstructions and Teleconnections. I work for a group called Past Climate and Ocean Studies at the National Center for Polar and Ocean Research. Given the diversity of the audience, my intention is to give some intuition into polar climate variability and why is it important, and how NCPOR actually facilitates this kind of research as a platform. And also, if there is still some time, I can give some high-level summary of some of the research in this area that I conducted at NCPOR. So polar climate variability is huge. In fact, some of the most visible changes of climate change actually happens in the polar regions. And there's a nice property that the amplitude of these changes grows with the time scale. So at longer time scales, these changes are much, much higher. So if we look at the difference between modern day we have just one ice sheet in tiny ice sheet in Greenland, single ice sheet in the northern hemisphere, and what it was some 18,000 years ago when vast expanses of the continental region was covered in ice sheet, and also some vast expanses of regions covered in sea ice, which is kind of diminishing here. So in a way, I would argue that Earth's climatic history has been a history of polar climate variability. And because these regions are so well connected through oceanic and atmospheric pathways, they tend to, the changes that happen in these regions also tend to form remote climatic linkages with distant climate subsystems, what we understand as teleconnections. So now, so if we look at some of the tipping elements in the Earth's climate system, which basically means that some of the elements that are vulnerable in a way that there could be irreversible changes to those systems, these are again mostly concentrated in polar regions. And we were not supposed to see this because on our time scales that we are, on human time scales, we were not supposed to be worried about polar climate change because these takes place over much, much longer time scales. But here we are that we have these tipping points. And the reason for this is this chart, which appeared as figure 1 in the latest IPCC technical summary report. What it says is that the climate change or the amplitude or the amount of change that is supposed to happen over millions of years is now projected to happen in the next 300 years. So these are projections, various projections of the IPCC, business as usual, some moderation to economic activity, and some drastic steps. But still, these are very, very large changes that are happening over very short amount of period. The Earth and our system is not very used to it. It has never happened. So again, although these changes are most visible or most conspicuous in the polar regions, but if we look at the global population density, we would see that it is mostly concentrated in lower latitude regions and also in the eastern hemisphere. And this is a region which is dominated by climatic system which is called the monsoonal climate. And we can see that how global population matches with the global monsoon system, which is, and the way it is defined is that a major chunk of the precipitation or other forms which defines monsoon actually is concentrated in a particular season. So it makes sense to study polar climate variability and also investigate how it teleconnects or how it forms remote linkages with monsoonal systems. So what really enables this kind of a study where we reconstruct climatic history, which are beyond the instrumental observation, is that we have geological archives, which are marine sediment cores, lake cores, speleothem, which are cave deposits, ice cores, tree rings, and corals. So a common theme between all of these is that these are layered and are sequences in time. So I personally work on sediment cores, and this is a close-up of a sediment core, where we, if we can, and because these are layered sequences in time, if we can date it, and we can analyze some of the properties going down core, we can actually build a climatic history using these materials. So our group is involved with studies, and these are the locations of some of the sediment cores which are spread across globally. And you would notice that these are some of the locations which are highly inaccessible. So NCPR really serves as a platform which provides access to these kind of materials. And it is a nodal agency or the Nodal Center for Multidisciplinary Scientific Research in the polar region. It operates bases in Antarctica, 2 bases in Antarctica, which runs 365 days a year. It also operates bases, a base in Arctic, which used to operate only during summer, but from this year onwards, it's going to be a year-round base. And also we have stations in Himalayas. We also conduct scientific cruises into the Southern Ocean. And we also partner with International Ocean Discovery Program, which provides a platform for going much deeper in time because of the kind of the drilling setup that they have. So this is our station in Antarctica, Maitri. This is Bharti. These are twin stations. This is our station in the Arctic, which will be operating 365 days from this year onward. This is our station, Himans, in the Himalayas. And also in the Southern Ocean, we conduct scientific cruises that goes every alternate year. And why this is important? Because if you see the global distribution of sediment cores, you'll see that the Indian sector of the Southern Ocean and the Southern Indian Ocean, some of the regions with the least data density, and this kind of cruises actually enables to sample from these regions. So also the International Ocean Discovery Program, NCPR, is the nodal agency for Indian participation in International Ocean Discovery Program. Just 3 weeks back, I was in Greenland sampling alongside these participants, where we drilled materials that were 2 kilometers deep inside the seabed, and our objective was to really reconstruct how what kind of configuration changes in the Greenland ice sheet in terms of geometry took place over the past 30 million years. So NCPR truly facilitates this kind of result. Now coming to some of my own work, I work in the Arctic region as well as the Southern Ocean, and I work on polar climate reconstruction as one, and also I look at how it connects to our climate system, which is dominated by the annual monsoon circulation. So starting with the Arctic, if we see the Arctic region is changing rapidly, and the evidence for that is the Arctic, something called Arctic amplification, where the Arctic region is on a whole is actually warming at twice the global average. But even within Arctic, some of these regions are warming at a much, if we look at the local amplification, are warming at a much higher rate than just a factor of 2, going all the way up to 5 or 6. And our sampling site is one such location, is Svalbard, which is currently some of the regions with the highest warming rate on the planet. So now, again, this region is a fjord, and what do we mean by fjord is basically it's a delta kind of a thing, but instead of land, what we have is glaciers terminating into the oceans. So before we could go deeper into the time, we conducted studies that demonstrated the climatic sensitivity of this region, where we demonstrated a steep biogeochemical gradient across this fjord arising from this glacial marine contrast. And under climate change scenario, these gradients could alter, which could get recorded as temporal signatures of changing paleo-environmental conditions. So with this context, we conducted one study where we built up a climatic history of this region for the past 1,000 years, and then we also investigated how it kind of resembled with the history, the variability of Indian monsoon precipitation. So what we did was that we measured certain parameters. These are routine parameters that we measure in sediment cores typically. We measured carbon isotopes, nitrogen isotopes, and organic matter elemental concentrations. But what I did was that we used some data detection techniques to cluster these into 2 sets. But we still did not understand what these clusters mean. So, and once we plotted this across the original parameter space, we found that a particular kind of cluster, which is represented by violet here, was absent between AD 1450 and 1850. So this was interesting, but we had to find out what was going at a physical level. So we took an independent data set, which is the last millennium reanalysis. It's kind of a hybrid between model simulations as well as all the proxies records combined into one, and was independent from our own data. And we created composites for our clusters, intervals that we got, cluster 1 and cluster 2, and then we took a composite difference. And we observed that the highest anomalies appeared around the same location where we had sampled. And because these were surface air temperature, we could categorize these clusters as warm and cold types. So we could generate this stripe, which basically the blue region shows cold intervals, and the beige in between is the warm intervals. So how would basically Indian monsoon respond to such a strong signal coming from the Arctic? For that, once we found that the cold Arctic intervals during the last millennial period, or the last 1,000 years, were associated with relatively lower precipitation over large parts of the subcontinent, and that we saw using both proxy data, where this is our record from the 1,000-year record that we reconstructed at our location Svalbard, and we also compared with the Indian monsoon record from marine as well as terrestrial regions. And we also looked at the composite difference between precipitation, and that also kind of matched. So colder Arctic conditions led to less intense phases of Indian monsoon and vice versa. Now moving on to the Southern Ocean, We used a sediment core from the subtropical front region in the Indian sector of the Southern Ocean to build a record of quantitative climate change over the past 40,000 years. And then again, we investigated millennial scale coherence between Indian monsoon variability and the surface air temperature at those locations. So what we did was that we measured magnesium by calcium ratios on carbonates, which using appropriate inversion schemes was converted to sea surface temperatures. And because we had quantitative data, we could also compare it with one of the few available simulations, transient simulations during that period. It sort of matched. We could see that the temperatures were lowest during the last glacial maximum and follows an Antarctic style deglaciation. And then we have a Holocene decline. It sort of matches with the only available simulation during that period. So we then compared our record of sea surface temperature in the Indian sector of the Southern Ocean with Indian monsoon record, both during deglacial as well as during MIS3, which is from 30 kiloyears to 50 kiloyears. And we observed that there was a near one-to-one coupling between millennial scale oscillations in Southern Ocean SSTs and the Indian summer monsoon. You could see how closely they tend to match. So higher SSTs in the southern mid-latitude regions resulted in weaker Indian summer monsoon, and lower SSTs resulted in a stronger summer monsoon. Now, why do I say millennial? Because if we look at the deglacial period, these two plots shown in black are the surface air temperature at our sampling site in the Southern Ocean, and the ones shown in blue are the cross-equatorial pressure gradient, basically surface pressure gradient between the Mascarene high region and the northern Indian Ocean. So we see that while they are correlated on the orbital scales, but on the millennial scale, they tend to anti-correlate and follows what we have shown here. And during period MIS3, which was devoid of any strong orbital trend, we could see how closely they are sort of anti-correlated. Now, that was fast. So coming to the summary, currently the tropical Indian Oceans dominate the year-to-year variability in Indian monsoon, from things like tropical Pacific, ENSO signals, IOD, even the equatorial Atlantic regions. But this could change in future as the polar climate variability grows and some of those thresholds are broken. So warmer Arctic coupled with diminished sea ice extent may intensify Indian monsoon in near term. And also we found that the southern mid-latitude SMLSs, southern mid-latitude sea surface temperatures was a predominant factor in controlling millennial scale variability in Indian summer monsoon. So with this, I would like to thank you all for listening. This was a very concise summary kind of a thing.
Thank you, Dr. Vikash Kumar. We do have time for questions on this very important topic. Yes.
Don't belong to this area at all, but I'm trying to understand what you are trying to do. Are you trying to correlate, or are you trying to find causal relationships?
So causal relationship is a different ballgame altogether. We do not have, it's correlational.
And so is it not possible that when you show these graphs, which have similar patterns,
yes,
is it not possible that there is a third set of variables that's affecting both?
Yes, it's quite possible. But we are limited because the kind of work that goes into generating that a ship goes, it drills, and then creates one record. So you can imagine if we have to, even if from a theoretical standpoint, we have some thing that we can kind of to understand the mechanistic links, it's still very difficult to do that.
All right, so what are you trying to do? Are you trying to estimate these correlations and then come up with a prediction equation? In which case, which is the dependent variable and which is the independent variable?
So the dependent variable, for example, if you look at Indian monsoon variability, the year-to-year variability is only 10%. But then on longer time scales, it's much larger. So what is really causing those variabilities? It's not, so it is coming from regions that are far away from that place, and that has to be the polar regions, which has those wild swings on those time scales.
But so you use the word cause, and that's where,
we do that, sir, but unfortunately we are very limited in our ability to handle causal relationships.
Please.
Okay, very interesting. What about greening of the Arctic? You know,
Yes.
netting and greening. Are you seeing similar? I mean, and what might be the consequences of?
The Arctic would eventually green if it gets rid of frost, and also there is a lot of widespread melting. It would green, but how would it relate to the work that I do? Monsoon, no idea. But it would definitely green. But at the same time, some of the regions which so in a sense it would affect the albedo. So when you change the greenery on the surface of Earth, when you remove the white patches with green patches, the largest consequences that are going to come is to the way we reflect the sunlight from the Earth, and that would change the radiative balance. But there must be modeling studies that take care of this. I don't personally do that.
Okay, thank you.
Any further questions? Okay, we thank Dr. Vikash Kumar once again. And I hand over the mic. The session is.
Oh, okay. Thank you. Thank you very much, Professor Chadda, for chairing the session. So now we come to the final talk of our 89th annual general meeting. And for this, the chairperson is Professor P.C. Pandey, and I hand it over to him.
Thank you, madam. Thank you. Good morning, good afternoon to all of you. So this is a very special lecture by very well-known physical oceanographer of the National Institute of Oceanography, who is the chief scientist, and a Bhatnagar awardee. His main area of dynamics of circulations of the ocean, air-sea interactions, and its connection with the monsoon. And this lecture, I congratulate the Academy to have thought of this very special lecture, because the period 2021 to 2030 has been declared by the United Nations as the International Year of the Ocean. And as many of you may be knowing, the government of India has launched a very ambitious program of deep ocean mission, providing 6 different themes or pillars to focus the research during the next decade. So in that context, my compliments and congratulations to all the academy people who has well thought of having this lecture. So Dr. Shankar, I request start your lecture. The topic is evaluation of or understanding of the dynamics of the vast Indian Ocean. As you know, this is a very old institute and was only institute till LCPR came into existence about 2 decades ago. So there is a lot of research and international class research work has been done about our Indian Ocean, about the physical, chemical, biological, and all other properties of the ocean. Very well studied and continue study by means of the ships. And now new technology has provided another avenue for getting the observation. So I think this lecture will be very interesting. Shankar is a very good orator. I do not want to talk. Shankar is my old friend. So thank you, Shankar.
Thank you, Professor Pandey. Am I audible? Because I can hear some. Okay, fine. So I'm going to talk about the evolution of our understanding. I mean, how we understand the dynamics of the North Indian Ocean. That includes the equatorial part. I'm going to try to step you through about 50 to 70 years of history with a few critical slides. At the end, I hope there'll be more questions than I have answers. So it's broken into 4 modules. In the first one, I look at the observed circulation and the theoretical explanation for the global circulation, not the North Indian Ocean. Module 2 looks at the largely linear theoretical framework we have used to explain what we see in the North Indian Ocean. Module 3 looks at the effects of nonlinearity, and Module 4 looks at the implications for prediction and insight, both of which are of interest. So the observed circulation. Now this is a map not very different from what Pai picked up when he was cast adrift of the Philippines. What you see here if you look at the global circulation, except the North Indian Ocean, is gyres. The most famous of which is in the Atlantic, and this is an intense boundary current on the western side. It's called the western boundary current. Here it's the Gulf Stream, and of Japan it's the Kuroshio. So you have semi-permanent subtropical gyres. The central latitude would be around just south of California perhaps, and there are intense western boundary currents. The Gulf Stream, for example, carries about 100 million cubic meters per second. So that's the kind of transport we're talking about, and they play a very important role in our climate system. Now, how do we study this? It's basically fluid dynamics as applied to geophysical flows. The scales are very different. If you consider the ocean to be a tank of depth d and length l, d is of the order of a few kilometers, about 5 km, and the length l would be about half the globe. That's the Pacific. So you're talking of something like 15,000, 20,000 kilometers. So if I were to represent the ocean basin by the length of this line, it's to scale. The depth of the ocean is the thickness of that line. So it's a very different kind of flow that we are looking at compared to what we see in an IC engine or the flow over an airplane wing. It's a thin fluid, it's a flow on a sphere, and the sphere rotates. We are sitting on that sphere. We are making all our measurements with respect to this non-inertial frame of reference, and we want all our answers with respect to that. I want, for example, the wind relative to the rotating Earth. So I don't have a choice. The Coriolis force is a necessary pseudo force. I have to account for it. And it has consequences. Both of them. Rotation and sphericity, I'm not going to bother about stratification. So here what you have is a slab. This is the wind blowing on the surface. So if wind blows on the surface, you expect the current to go downwind. But that's not what happens in the surface of the ocean. The current is directed at right angles to the wind. Because the moment the wind starts moving the water, the Coriolis force sets in. It comes into action only when there is motion. The Coriolis force sets in, the wind balances the Coriolis force, and you have a flow at right angles. So if I have the wind blowing this way, the current goes at right angles to the wind in the northern hemisphere. Now this is called Ekman flow. This is one bit of jargon I will use a bit as we go along. The other consequence is if you look at a tilt of the sea surface. So you have high pressure on this side, low pressure on this side. As you expect, the current starts flowing down the pressure gradient, but then the Coriolis force sets in and the current goes into the wall. So again, it's at right angles. If this seems surprising, just imagine what you see in a cyclone. The low pressure in the center and the flow is around it. So it's not something we are not used to. This is called geostrophy. This is the other thing that's of interest. So you have Ekman flow, where wind stress balances the Coriolis force, and you have geostrophic flow, where the Coriolis force is balanced by the pressure gradient. Now this leads to some remarkable consequences. This is steady state. But if you look at time-dependent circulation, you can do a full course in mechanical or aerospace engineering without encountering waves, except shock waves. But that's not true of the oceans or the atmosphere. Oscillations dominate. So you are looking at waves from word go. So this is a summary of the global ocean. You have these gyres. You have a very thin fluid. You're looking at sphericity and rotation. And you have these subtropical gyres. You have intense western boundary currents. The explanation for why you see this circulation in 5 basins, North and South Atlantic, North and South Pacific, and the South Indian Ocean, came in just 5 years following World War II. 3 remarkable papers, Sverdrup, Stommel, and Monk. And these were steady state solutions because these gyres exist throughout the year. They were steady state solutions, and what you see is westerly winds in the mid latitudes here and easterly trades coming towards the equator. So that's an anticyclone. You are putting in angular momentum, and that's what leads to these gyres. Why do you have the western boundary currents, not at the eastern end? You have them at the western boundary because of the directionality introduced by the Coriolis acceleration. It goes from negative maximum at the South Pole to 0 at the equator to a positive maximum at the North Pole. So there is a directionality, and that shifts the boundary current to the west. Now these were steady state theories. When the Challenger expedition mapped the oceans in the 1880s, they bypassed the North Indian Ocean because it was too small to be looked at. Here you have reversing winds, and that was the inspiration for the International Indian Ocean Expedition in the 1960s. It was a remarkable international program with a very large number of countries participating. So you have the winds here in January, the northeasterlies and the southwesterlies in July. That sets up a reversing circulation. So the question is, how do you look at the circulation and what exactly do you see? Now in this short talk, I need to focus. So what I'm going to do is look at just the circulation of the coasts of India. But as we shall see in the second module, to explain even what I see of Goa, I need to simulate the entire basin. So my institute was set up following the IOE and the liberation of Goa in 1960s. What I look at are the results of some cruises in the Indian EZ from 1987 to 1994. And they were carried out by my mentor and academy fellow Satish Shetty and his colleagues. So they were very systematic hydrographic observations. There were 8 cruises spread over 8 years, and they inspired theoretical studies. So finally you had a set of cruises that mapped the circulation and told you what's happening to the currents of the coast of India. And that led to papers that could use models to simulate these observations. So what happens along the west coast of India? Now you look at these winds, they reverse with season. But there is an interesting catch here. What is of interest to us is the alongshore wind. Because what happens is the following. Suppose this is a coast. This wall is a coast, and I have a wind blowing away from me. Okay, so it is with the coast on its left. What happens is you have Ekman flow. It moves the water out. So the thermocline or the isotherms come up and the sea level drops. Once the sea level drops, you have a pressure gradient with high pressure to my right, that is offshore, and by geostrophy a current sets in downwind. So that's what happens if you have an alongshore wind. If you have a cross-shore wind, on the other hand, it barely sets up a longshore current. So the direction of the wind matters. Off the west coast of India, the wind is mostly directed normal to the coast. So if you look at the component along the coast, it is equatorward throughout the year. The West India Coastal Current, or WICC, flows with the wind, favoring upwelling during July, during the summer monsoon. But during the winter monsoon, it flows into the wind. So this is something we would like to explain. Why do you have the current flowing into the wind? It's more easy to understand why the current flows with the wind. That's the normal expectation. Off the east coast, the geography is such that the southwesterlies are almost along. So most of the wind, though it is weaker than in the Arabian Sea, goes into driving a strong current. But curiously, it is during March, April, when the winds are at their weakest, that you have a strong boundary current of the east coast. Now, these are practical implications. If you are drilling for oil in the KG basin, you need to know what the currents are and what the winds are, because both move your platform. You are sitting on sea surface, which is not exactly stable, and you are drilling at the end of about half a kilometer. That's where your drill bit is. So the stabler your platform, the better your chances of drilling. By July, when the wind picks up, the current collapses. So what explains what we are seeing? Now there are questions that I've listed, but I'm going to look at just one of them. Why does the West India coastal current flow into the wind during the winter monsoon? The basic theory is contained in 2 papers by Taro Matsuno and James Lytle, and a PhD thesis unpublished, Dennis Moore, 1968. All of them dating back to the 1960s. Matsuno identified the equatorially trapped waves. These are based on the Hermit functions. So these are the dispersion curves of the waves that you get on the equatorial beta plane. Lytle recognized that there are waves called Rossby waves which propagate westward, and they propagate faster near the equator. If you go to the latitude of Los Angeles, you will see it going at 1 centimeter per second across the Pacific. which means it's going to take about a decade to go from the American west coast of California to Japan. If you come to Kochi, it's 10 centimeters per second. So you can have the Rossby wave going to Somalia within the season. At the equator, it's about 60 centimeters per second. So there's a huge difference between what you see in the mid-latitudes, where it's very slow, and what you see at the equator. This is what Lytle showed. Dennis Moore's thesis showed how to reflect what are called equatorial Kellen waves. They go eastward. I'll come to that later, at an eastern boundary. So okay, now here you have a rectangular basin. This is an analytic solution from Julian McCreary, his PhD thesis. What you have here is a rectangular basin. This is in kilometers. And you have a patch of winds restricted to just this part. So you have east-west winds, so they are directed this way, and they decay away from the equator. So you have a Gaussian that decays away from the equator, and again it decays as you go towards this edge. So it's basically restricted to this patch. The response, however, is not restricted to that patch. The waves that are generated, they take the signal created by this wind patch eastward. That's called a Kellen wave. And it goes along the equator at about 2 meters per second. So in a few months, it's at the other end of the Pacific. It reflects as a Rossby wave. Now look at this curve, this parabolic shape. The speed of the Rossby wave goes as roughly 1 by y squared, where y is the distance from the equator. So you have a high speed as over here at the equator, and very slow once you're away from it. So that's why it takes about 10 years to go across the Pacific at Los Angeles, while it's going to do that in a matter of months at the equator. But the reflection is not restricted to just the Rossby wave being produced here. You see a coastal Kellen wave also going poleward on both sides. So these signals can be tracked to Alaska and to Chile. So there is actually a paper in Nature in '93 by Jacobs et al. They used the then available GeoSat altimeter to track the impact of the 1982 El Nino, which was the biggest on record till '97. And they tracked it right across the Pacific. So you could see this equatorial Kellen wave and the coastal Kellen wave. This is over within months. But then the coastal Kellen wave pinches off the west coast of the US and then moves right across the basin. 10 years later, it is seen affecting the climate of Japan, the weather of Japan. So that's a kind of memory that you have. Now, this is called remote forcing. I have winds only in this patch, but it affects a much wider expanse of the Pacific. In fact, practically the entire Pacific if you wait long enough. So why does the West India coastal current flow poleward during winter? The first explanations that came invoked salinity. So you have low salinity here, high salinity here. This is the Bay of Bengal with a lot of rivers and a lot of rainfall, a lot of evaporation, and the Indus is almost completely dammed. So there's very little here. So this kind of salinity gradient you don't get anywhere in the world oceans over this kind of distance, except at the mouths of rivers, but they're highly localized. Such a systematic gradient you don't find anywhere else. So the first theories looked at the low salinity here versus the high salinity here and showed that you could actually drive a current into the wind. But if you look at purely dynamical models, these don't have any horizontal variation of temperature or salinity. They produce the current going into the wind in winter. And you need the pressure gradient. You have a high in sea level here. The colors show you sea level. So you have high sea level red, and blue is low sea level. So you have higher pressure here, lower pressure here, and you drive a current into the wind. What produces these pressure gradients? It's not salinity. We don't have salinity in this model. We don't even have horizontal variations of temperature. It's coming from this Kelvin and Rossby waves. So you can actually show that by splitting the response. This is a simulation with only west coast winds. You see a response, but it is weak. This is the full response, and this is what you get if you have only west coast winds. That's because the wind is strong, but it is almost entirely blowing into the coast. You have a very weak longshore component. While the east coast wind is just an accident of geography. You have southwesterlies and you have a coast that is perfectly oriented to put almost the entire wind alongshore. So you have a strong response of Kerala coming from the east coast winds. This is a simulation with only east coast winds. So what happens is you're generating Kelvin waves that go around here and then radiate off as Rossby waves. So remote forcing from the Indian east coast is important for the seasonal cycle of the West India coastal current. So if you're interested in explaining what happens here of Goa, you need to simulate the entire basin. And the problem is it is not restricted just to very large scale systems. Even if your measurements are at something like 10 to 20 meters water column depth, which is where environmental impact assessments are typically conducted. You're looking at the impact of effluent pipelines. The result is the same. So you're talking of a system which has a very large scale, even if you're looking at a very local problem. The simplest model, you just sum the Hermite functions. These are the basis functions for the equatorial beta plane. Now, this is the kind of picture that Chettier brought back when I first came to NIO in '95. I mean, these were on transparencies. The older people here would know them. 12 transparencies, one for each month and global. But even in a global picture, what was striking is this sea level signal that moves off the west coast of India. It's very striking, even in a global transparency, which is just A4 size. Now, this was the first time, this was around 93, 94, that oceanographers could look at a near synoptic view. The repeat cycle is 10 days, but it is as good as synoptic for us. In those days, the processing techniques could give you about a monthly picture at a coarse resolution of about half to 1 degree. Today, they do much better. So how do we explain the observed circulation? You have a waveguide which is leaky. The red is the coastal waveguide and the blue is the equatorial waveguide, and energy leaks from one into the other. So if I want to simulate the current of just Goa, I need to simulate this entire basin. That is the catch. And that's because remote forcing is important. You produce a signal here, after some time it has an impact somewhere else. And the nature of the waves depends on what the system permits. So that's module 2. This was entirely linear stuff. But we know that fluid flow is nonlinear. So what happens when you start looking at nonlinear effects? This is an example of nonlinear dynamics. This is the coast of Oman. You have the southwesterly winds during the summer monsoon blowing this way. So what do you expect is smooth Ekman drift offshore. That's linear theory, classical linear theory. But that's not what you get. This is a chlorophyll map from satellite. And what you see is that the Ekman flow is not smooth offshore. It's in fact concentrated in squirts and jets that are associated with these capes. If you take a high resolution general circulation model, this is at the resolution of about 5 km, you can reproduce that kind of a system. But you will never get this with a linear model. So there is a nonlinearity here that needs to be accounted for. General circulation model basically divides the ocean into stacks in the horizontal and vertical and solves the primitive equations. They are nonlinear, and these are solved in finite difference form even today. So what is a nonlinear system and what is a linear system for those who are not familiar with that? Suppose you have a black box here. That's our system. And you have inputs I1 and I2 giving you outputs O1 and O2. So I push I1 or I2 in and I get O1 or O2 out. Now if I put in the input I1 plus I2, a linear system will give me O1 plus O2. That's by definition a linear system. In a nonlinear system, you're not going to get O1 plus O2. What it implies is that the principle of superposition, which we learned in school in physics, is valid for a linear system. It's not true for a nonlinear system. Now this is the way the derivative looks for any property S. You can have a local change with respect to time and a change that depends on motion. So let's just look at it quickly. Suppose you have a constant temperature in this room, but you allow for spatial variation. I mean, you will find it is colder near the AC vent than it is away from it. So you have a constant temperature, not changing in time, in that if I put a probe at any location in the room, I'm not going to see a change in time. But suppose you have a flow of air, and it's going from some temperature T1 to a temperature T2. By the time the parcel goes from here to there, its temperature has to change from T1 to T2. Now that change comes because of this multiple of the speed with respect to the directional derivative. Okay, so this is called advection. So you can change the temperature either locally by, say, fluxes, SE fluxes or whatever, and you can change the temperature simply by advecting a parcel. This is nonlinearity, and it's fundamental to all fluid flows. Nonlinearity also happens to be an essential property of what is called complex systems. With complex systems, you get self-organization into patterns. You just look at any weather forecast, you're going to see cloud patterns. Massive cloud patterns organized on a variety of scales. You start looking deeper into that, you will see more patterns. So there is self-organization into patterns. There is chaotic behavior. I'll look at chaos briefly. There's fat-tailed behavior. You get many more extremes than you would expect from linear theory. There is emergence, which is a consequence of nonlinear interaction. These are properties that emerge as a consequence of nonlinearity. Now, these complex physical systems are studied using partial differential equations. There's a fairly well-established body of mathematical theory for that. The problem starts when you start looking at systems that are not amenable to that tool. If you look, for example, at certain aspects of ecosystems or stock markets, because of adaptive interaction, the system can adapt, and then the theory of partial differential equations breaks down. I'll look at chaos and emergence very briefly. So these are the three equations solved by Edward Lorenz in his 1963 paper. They represent classic convection as in the atmosphere, heated from below and cooled at the top. Very simple equations, but they gave rise to this kind of behavior in phase space. Now, the system is bounded. Your variables don't blow up in time. But there is a limit to what you can predict. The horizon is limited. In the atmosphere, for example, you can't make a sensible prediction beyond about 7 days. So you will not find a weather forecasting model that goes beyond that. So this is chaos. You see it in the oceans. My colleagues launched 3 drifters. These are buoys that you just throw out into the sea and they drift. They are Lagrangians, and you can track them using satellites. On 29 November 2016, out here, this is Gujarat, so this is the domain of the West India coastal current. They just dropped them off, and these are just thrown out within minutes. So that's instantaneous as far as we are concerned. They went together for about a week, but then a few months later, they were found in 3 different parts of the Arabian Sea. So that is something you cannot ignore. And this has practical applications. With COVID, we have forgotten one of the most remarkable disasters in aviation history, MH370. It disappeared on 8th of March 2014. I had a difficult time explaining to my wife why in this age, 2014 was bad enough, but in 2023 it's even worse. Why you can't find the debris, why you can't find out where it went down. So this is the rough arc. There was only one sensor working in the engine, so it's a scalar. So you can't pin down where it was, but you can pin down a rough arc where it might have gone down. A few months later, off Reunion Island, they found a wing part. So that is the South Equatorial Current, which is the northern arm of the subtropical gyre in the South Indian Ocean. That's classic linear theory. But these are the drifter trajectories from 1985 to 2015. This is chaos. It tells you why you can't find the place, why you can't find the plane. It's a huge area. It looks small out here, but if you look at the distances covered and it's 5 kilometers down, it's an impossible task. It's worse than looking for a needle in a haystack. I really felt sorry for those hydrographers who were sent out on this job. They knew they were guaranteed to fail. Yet there was no choice. They spent months out there at sea. I mean, every day you wake up, you go out, you know you're guaranteed failure. There's no hope, but you still have to do it. Okay, that was chaos. These are propagating waves that we saw. Now you can see them at intraseasonal periods, that is within a season. You can see them at the seasonal time scale, which is the most dominant cycle in the North Indian Ocean. You can even see them at interannual periods. So these Rossby waves that pinch off Kerala or off the equator here and just go westward, they exist at all these time scales. So this is a map, a fast Fourier transform, showing you the amplitude at a period around 3 years. So it's an interannual band. It's a period that you would associate with probably El Nino. Not quite, but it is a rough band. What it shows you is the amplitude of the FFT. So red is where there is high amplitude or high variability, and blue is where you have low variability. So this is a logarithmic scale, so every 1 unit here is a factor of 2. So between this blue and red, there's a factor of about 100. Now you would expect that if you generate a Rossby wave out here that has such a high amplitude, you should see its impact right across the basin. The question is, why don't you see it? There are regions here where nothing seems to happen. It's just static. You can use a very simple linear model to simulate this. You can actually get an analytic solution if you replace India by a knife edge. You can do it with paper and pen. And you produce practically this observation. But look at one aspect of the linear simulation. It's smooth, as you would expect for a linear system. And of Oman, the linear model produces a very smooth minimum. In the observations, you have blobs. You have broken patches. If you run an oceanic general circulation model, fully nonlinear, primitive equations, at a reasonable resolution, you get those blobs. I mean, you run it at an even higher resolution than down here, you're going to see much more cleaner comparison with observations. So this is the impact of, this is emergence. And why is it happening? Because of Oman, you don't see that smooth Ekman drift that the linear model picks. You are seeing these kind of squirts and jets that you get in a nonlinear system. But it is the instability of this linear current along the coast of Oman that is producing these blobs. So the current goes unstable, and it goes unstable producing the squirts wherever you have these capes. But if you take a linear system, the capes basically disappear as small perturbations, and you get a smooth flow offshore, producing those kind of minima. So this is emergence. This is a property that arises as a consequence of nonlinearity. So module 4 looks at the implications for two things that are of interest: prediction and insight. This is insight. It tells you why it happens. There is this Rossby wave, it does go across, but in these regions, there is a local forcing that cancels it. And the Rossby wave is linear, it re-emerges at the other end of this local forcing patch. You see its effect again. So with this model, I can understand why we are seeing what we are seeing. This is simulation. This is accurate. I can't get that kind of thing with the linear model. But if I were to run just this 5 km resolution GCM, I would not have any idea of what's actually going on. So you have simulation and you have insight, and the two need not go together. There is no question of why you want prediction. That's obvious. The chlorophyll filaments can be simulated fairly well with a high resolution model forced by observed winds. Now in a forecasting system, the only way you achieve reasonable accuracies is by what's called data assimilation. So you look at a typical atmospheric forcing model, atmospheric forecasting model. You run it for say a day, then you take in the data that Professor Atmanan spoke about from various sources, land and sea, and you assimilate them into an analysis, and you run what's called either an adjoint operator. When the model runs backward and forward, it tries to assimilate the observations, blending the observations with the model. So you're basically getting a cross between what the model tends to simulate. It drifts away from observations, so you're getting a model state combined with observations. So it's neither model nor observations, it's a cross between the two. And that's the only way you get reasonable accuracies in a forecasting system. and its relevance becomes less and less as you go further into the future. So the chaos that is inherent in such nonlinear and complex systems limits the forecast horizon. In data assimilation, the model is run for some time, and you assimilate data and you rerun it. So every day you see a forecast being updated by the India Met department. Now what do you do when you want to simulate climate? This approach is not viable. You can't assimilate data. There's nothing available to assimilate over 70 years. What you do is throw in scenarios, carbon dioxide, whatever, this scenario or that scenario, and you run it, assuming that that's going to be constant. So you cannot assimilate data when running a model over a season, which is what IMD wants to do, make a seasonal forecast, and it's difficult because it's purely model dependent, or years or decades. Yet forecasts are made, and there are serious practical consequences of any decision made on the basis of these models. So what's the difference between simulation and understanding? Simulation implies capturing as much of the system's complexity as possible. Understanding implies sufficient simplification to capture the essence of the phenomenon. This gap between simulation and understanding was discussed by Isaac Held, an American metallurgist, in an essay in 2005. The practical value of simulations is undoubted. Nobody doubts it. You want to make a prediction. I mean, the stock market reacts to monsoon forecasts. But in today's world, is understanding or insight still important? I'm going to leave you with this question, though I try giving an answer to it. Held notes one practical importance of understanding, comparing models. Now, if you look at any IPCC forecast, for any given scenario, you are starting off all the models from the same state, same set of initial conditions. They diverge after some time, and there is a huge spread. You have, say, 15 models, and you have a huge spread in what they say is going to happen 50, 70 years from now. How do you know what is right and what is wrong? So that is one practical importance of understanding, comparing models. You want to know why a model goes wrong when it goes wrong, and you want to know why a model, for that you need to know why it is correct when it is correct. And I recall one statement by the former Pakistan cricket captain Imran Khan. He once said, good teams analyze their failures, great teams analyze their successes. So you want to know what is causing that spread. Which one do you believe? And there are huge practical consequences. Do you buy an electric vehicle or petrol vehicle if you are going to replace your vehicle tomorrow? The problem lies here. Now, climate constitutes a complex physical system. Isaac Held compared climate science with molecular biology, which also deals with complex systems. But he noted one vital difference. There's a complete hierarchy of biological systems, from viruses and bacteria on one end to human beings on the other. You have the entire spectrum available to you. There is no such hierarchy of climate systems. So you have to construct a mental hierarchy of these climate systems if you want to do what biologists have been able to do. Held compared the phenomenal progress in molecular biology over the last few decades with the relatively poorer progress made in climate science. He also noted one more thing. Though biologists have an incredible variety available to them, they still focus on one or two organisms. E. coli, for example, and Drosophila melanogaster. You will find an incredible number of papers on them, because if you study E. coli and I study E. coli, the results are comparable. So Held said that you actually need some focus. You cannot build, everyone cannot have his own model. You need some consensus. And this consensus is not easy because you have national models, you have geopolitics, a lot of things coming in. So it's not easy to build this consensus, yet the consequences are global. So you need to build this hierarchy of models, and they are mental constructs, not like what you find in biology. So complex systems, you have this buzzword today, big data and models, and there are two competing views. They are very old. One extreme view is due to Fredkin and perhaps best expressed by Stephen Wolfram of Mathematica. The universe is in essence a simple program. A set of algebraic rules, you just start from some initial condition and let it spin off. And you can generate phenomenal patterns. This is not a cyclone. It's a fractal image of that looks like that. So is this how atmospheric science works? This is not broccoli. This is another fractal image. So does nature 3D print broccoli? So this is one extreme view. And that is what we are kind of heading towards with artificial intelligence. The other extreme view, which perhaps is no longer as strongly prevalent, is due to Clifford Truesdell, who called the computer ruin of science and a threat to mankind. This is a 1984 paper. But you have serious questions with the explosion in computing power and what is called proof. We had a talk some time back where proof is complete. You can probably find mistakes in the proof, but once it is proven, it's finished. You cannot disprove Pythagoras' theorem. You can find better ways to prove it, but you cannot disprove it. It's over. That is the way mathematics works. But here you're talking of proof in a complex system and one which has consequences for humanity, climate science, for example. So how do you deal with this? That's why this problem with proof is why you can take different positions with respect to climate. There are issues with no matter what model you pick up. But this competition is not new. In two articles in 2003 and 2005, Rohan Narasimha looked at the difference equations and accurate astronomical predictions that were made by Indian astronomers, and they astonished a Scottish mathematician called Playfair. He's the one who was taken aback by that. Till Newton and calculus, these predictions were far superior to anything in Europe. What they were doing is using difference equations. They were meeting every few decades, updating coefficients. They had no problems using epicycles. To Aristotle, it caused a lot of fundamental philosophical problems. So they were basically doing some kind of crude data assimilation, but they did not have a model. And they abhorred models, while Aristotle had models. And he had those concentric spheres, spherical spheres, concentric spheres with glasses. It was the marriage of Indo-Arabic algebra and Greek geometry with Descartes and the Newtonian revolution that changed everything. So model building started with that. The question is, in this age of the chip with Moore's law, big data, artificial intelligence, and self-learning neural networks, does the differential equation still matter? I have a serious question here because I have to make a practical judgment. In a limited curriculum, do I still teach the dispersion curves put down by Taro Matsuno? Or do I tell them just forget about it, just learn how to code AI? There are practical consequences, and I'm sure that I'm not alone with this question. So that's an interesting topic for discussion. So it's a question of calculation, as in simulation, versus deduction from axioms or first principles. It's a philosophical problem, and at the same time, one with very severe practical consequences. Just go back to Isaac Held. There's a practical, not merely theoretical or academic need for insight. You're projecting into the future with no way to know which of those models is actually going to give you the right thing. None of them succeeds with the current Indian monsoon, for example. And even with respect to something like aerosols, there are diametrically opposite schools of thought. One says that India will benefit, there'll be better rains, and the other says no, it's going to be a dry state. So what do you plan for? If you're an agricultural scientist, do you plan for floods, or do you plan for, what do you call that, droughts? My own view is take the middle path between the pen and the chip. I don't know how long it will run, and relevant data. The last point here is that we always think in terms of big data today, but earth science is a sparse data problem. If you look at the elements of the earth system as a matrix, there are parts which are very dense, which is where you have satellite data coming in. But there are large parts that are blank, and they contribute equally, if not more, to what you are seeing. So I will not go over this summary. Now these three pictures give you three views of linear versus nonlinear flow. Three measures of linearity. So where you have these purple and blue colors, you have a dominance of linearity. So you can see that in our region, in the tropics, except near the western boundary, it's fairly linear. It's a full toss and open goal. You have to put it away. So that's it. Thanks.
That was truly marvelous. I had a question about taking land masses into consideration in because your land masses and the atmospheric conditions of heating and are also then going to drive your wind, which
Correct.
will drive many of the phenomena.
Coupled system.
Sorry?
It's a coupled system.
It's a coupled system. So I was wondering how are land masses taken into this scenario at all, or that's not
It depends
relevant?
on what kind of model you're using. If you're using an ocean model, you take the winds as given. If you're using an atmospheric model, you take the sea surface temperature and the land surface conditions as given. Then there are coupled models. So when I was a student at IISc, they were very bad. This was in early 1990s. Today they are good. And that's what you're seeing in the IPCC reports today. How good they are, you judge by the spread. And you can take any view. So that's why in the US you will still have a strong political debate because both are plausible.
But is the spread declining over time with refinement of models and data?
See, what does the model do? It takes a certain view of the phenomenon and puts that into mathematical form and then codes it. In any atmospheric model, the problem comes with processes that are within a grid. So you have your grid nodes. Say you have a 50-kilometer resolution. So every 50 kilometers you have these points where you have your velocities, for example. What's happening within that? And the biggest problem is cumulus parameterization. The cumulus clouds that you see have a length scale of about 10 km. And they are turbulence. The other part of the code, an atmospheric forecasting code that eats up time, is radiation, line-by-line calculation of the radiative heat transfer. So in a way, those two problems that Kelvin noted in 1889 are still there with us. It's solved, the black body radiation problem, but radiative heat transfer is a nasty piece. It eats up a lot of computer time. So AI may help there in reducing the computer time. Cumulus parameterization, it's still a mess because you're dealing with turbulence at its core. It's still an unsolved problem. So I'm sorry, I don't have an answer to your question.
Shankar, you just said that artificial intelligence might help. I mean, the philosophical problems aside, there are some developments over the last couple of decades on using AI ML methods such as reservoir computing and so on that actually make it possible to do medium range predictions in chaotic systems. Have these been used in climate models at all?
I don't know. I won't be able to answer that. But I'll tell you the bigger problem is not which method you use, whether you use AI or don't use AI. The problem is not with method. The problem is the unwillingness to recognize that you are not dealing with a big data problem. You are dealing with a sparse data problem. I don't have a world map here. I'll have to go back to that slide. But if you have some idea of African geography, from the Red Sea, where you have Sudan, for example, and Eritrea, to almost the Atlantic on the other side, there are no radio sound observations. There's civil war and a mess all over the place, over a wide swath of latitude and the entire longitude band between Red Sea and this place. Now you stretch it into West Asia and farther north into Ukraine. Would you be launching radio sound balloons given the conditions out there in Ukraine and Russia? So huge swaths of places where you could actually make measurements. This is not like the ocean, where you can't make those radio sound observations. They're out of bounds today. You have either no money or you will probably get shot down.
If there is no
question at the end of your very beautiful talk. Uh, the question still remains is that since we started in 1980s, late 80s, about the physical oceanography cruises in the Bay of Bengal and the Arabian Sea, which I'm familiar with that, all those cruises. And so down the line in now 2020s, what is the new information which we have achieved in terms of the dynamics of the North Indian Ocean?
Okay.
Which we can say that this is what we were not knowing 30 years ago, 40 years ago, and this is what we know today, so that our prediction or understanding of the biogeochemistry coupled system, as well as the physical exchange between even you can say within the ocean or the air-sea exchange,
Okay.
whichever.
I have those slides, but
just a one-line answer which I would like to.
for the last 15 years, we've been running a program making direct current measurements. Now, we have enough data now to ask one very simple question. On a given day, let's say 5 November 2023, in what direction do you expect the West India coastal current to flow? And the answer is it could go in either direction. It is not the picture you got from hydrography. Intraseason. No, wait, let me finish. You go to the east coast of India. The old observations, the hydrographic observations of Shetty et al., they are still valid because the seasonal cycle dominates. So there is a greater probability that I will get the current in the expected direction based on my understanding of a seasonal cycle. On the west coast, it could go practically in either direction because intraseasonal variability dominates. Now, what this implies for biogeochemistry is a different business altogether.
Thank you.
Thank you so much, Professor Pandey, for chairing this. And I'm sure you'll all agree that we ended with a fantastic lecture which brought all the disciplines of science together. And I think we really delighted Professor Shankar to have heard you talk today. So I guess all things have to come, good things have to come to an end. And we are really sad that we are at the end of 2.5 days of delightful intellectual engagement. I hope you do agree with me. It's not good for me to say that it was delightful. All of you should agree that it is. And I'd like, on behalf of the Academy, to thank all of you coming here and contributing. Many people whom I have to thank. I hope I get all the people right. Of course, BITS Pilani for being our partner. Thank you very much, Professor Ram Gopal Rao and Professor Kundu. You've really given us a wonderful experience, even yesterday on the boat, which was really very nice. And of course, our other partner, NCPOR, we have a representative, Vikash, who gave us, lovely insights into what's happening in the Arctic and the polar regions. So thank you to both these organizations for partnering with us. Of course, I have to thank the staff, the administration staff of BITS Pilani, the Dean, Minal. Thank you so much for being, the LOC has been remarkable. Our little booklet contains all the mobile numbers of everybody that we could contact, which is quite unusual. And we really thank BITS Pilani and the administration and the entire LOC for being so marvelous. Thank you very much. And of course, I have to thank the Academy staff who have been working tirelessly behind the scenes, and Mr. Mahesh Chandra is trying hard to keep awake because he's so tired. Thank you, Mahesh, so much, and all the Academy staff who I'm sure have helped you with all your travel reimbursements and I don't know, all the other things that. Thank you very, very much. Thank you to the catering. Thank you to the taxi people. Thank you to everybody. I hope I haven't forgotten anything. Yes? Audio, video, yes, everything. And everything will come. And of course, on behalf of Vijay Mohan Pillai, who's secretary, co-secretary with me, but he's not here, and Raghu and Umesh, happy journeys and good lunch and safe journey back home. Thank you all.
Um, Please join me in thanking Professor Anne Borges, Vijay Mohanan Pillai, and Professor Raghunathan, who put in a huge amount of effort in the intellectual planning and various detailed aspects of this meeting. Thank you. And on behalf of the Academy, it's our pleasure to, yeah, okay. Professor Ram Gopal Rao and Professor Kundu. Uh, So thanks to all of you for being here in such good numbers and very active participation to make this a vibrant meeting. We look forward to having similar vibrant meetings in the years to come. Thank you, one and all.
Professor Umesh and Professor Borges' sentiments. Thank you very much for being here. We have always been a fantastic teaching institute. You know, we provide the best skills in the country possible to students. But over the last decade or so, research has been, thing that you're pushing hard for, and your presence will make a difference. Thank you very much for being here, and all our young faculties, really enjoyed the technical sessions and the learning lessons, the public lectures. So we are really grateful to you, and also an appreciation for our students. Many of our PhD students actually were involved in volunteering and helping us. Thanks to them as well. Please keep visiting us. Please keep visiting us, not only as in this sort of gathering, individually as well, for various purposes, talks and examinations and more so. Thank you very much. Thank you very much for being here. And also, if you are around, we are celebrating our 50 years. So it's 20 years. No, I didn't sleep. I didn't sleep last night. And it's starting at 2:30 in the auditorium. And we have also organized for an open day tomorrow, the entire day. In all our departments, divisions, student clubs, they're exhibiting their creations. If anyone is around and if you wish to come, please let us know. We can arrange for vehicle or whatever way to visit us. Yeah, I'm looking forward to 50 years. But thank you very much. And lunch is arranged. Some buses are here, so they can take you to the lunch site, or we can walk, 2.5 minutes away. Thanks. Thank you so much.